Okay. Hello everyone, good evening. Welcome back to our YouTube channel AgriAddict. I hope I'm audible to you all. So we'll be starting the session very soon, another 1 to 2 minutes. So just share the link with your friends who are all giving the ASRB net exams for the subject of seeds and technology. So this is like one short video for the complete seed testing, which is the one of the most important topic or the section for the ASRB as well as like ICAR PG, the PSD, all the agriculture exams. So no matter like whether you're giving the ASRB net exam, you're preparing for ICAR PG, PSD or any other state agriculture exams, state government exams, central government exams, this video will be a lot more beneficial and useful for all of you. So in this video, what we are going to do, so I'll be discussing about the content outline first, like what we are going to cover. We'll be mostly covering all the aspects of seed testing that you should be knowing about. Okay. And next, we will be also talking about the very important things that you have to remember and what are the proper source. See, what happens with the seed technology? There is a problem with the content where many content are given differently in the different books. And at the end of the day, we will be definitely confused about like which one to follow and which one to read, right? So that's what like we'll be also simultaneously explaining you like this content is taken from which source and which source you have to follow for said exams. Okay. So if there are any doubts, you can definitely post it in the comment section, but not about the PG rank because as expected, like the PG rank was again not given. It got delayed, and the students have, and also like we came to know that it will be postponed for another 1 week. So you can expect anytime in the first week of September, hopefully. Okay. So anything apart from that with related to the seed technology, you are always free to comment. Can we start? I hope like I'm audible to you all. Just give me a confirmation. Yeah, please say yes if I'm audible. Okay, fine. So let's start. So what we are going to discuss, I'll just tell you the basic things. Firstly, so if you are a student of seeds and technology, whether you are PG, PhD, or you are preparing for ARS, ASRB, SMS STO, when it comes to the seed testing, where you have to follow and what things you have to minimum read. See now, ASRB net exam for the 2025 will be connected in like few days, but if you can't read it now, like if you are a student who is watching this video, will be giving the exams in the upcoming years, so this will be very much useful. The first thing, very basic thing is just you have to read the complete ISTA manual. So this is mandatory. Second thing, you have to also go with the IMACS. So things with certification will overlap here. Third thing, third best source to go with is like RL Agarwal. And the fourth best thing that you can go for is like AgriAddict compiled notes. So that is available. Apart from this, still, if you want to read something else, something extra, you can always go for the other books like AK Joshi BD Singh, and you can also go for Karen Bale. Most of the things are like one and the same, but still, if you want to read it, you can also go for these two books. Okay, mostly these four things will cover entire seed testing, which is required for all agriculture exams. No matter what kind of question they ask, mostly it is confined to this. Okay, that's it. So this is the clarity that every student should be having, like where to read the seed testing from. So in this video, what we are going to discuss. So I'll just give you the brief outline on like what we are going to discuss. The first thing what we are going to see here is like we'll be talking about the purpose of seed testing, like why exactly the seed testing we are doing, and we will be also discussing about the world seed history, and next the Indian seed history, the details of ISTA, which is the one of the very important organization for the seed testing in the world. Okay, this is what we are going to discuss. Next, like ISTA accredited labs, what are these and how many are there in India? And next, what are the ISTA certificates and what kind of certificates are given by the ISTA and what do they mean, that we are going to discuss. Next, the ISTA recommended methods. See, consider this to be a most important one for your exam, where for the exam directly they will directly ask you about the ISTA recommended methods. Okay, for the vigor, for the viability, for the moisture, for seed health, like for all of these tests, the ISTA has recommended some of the tests to be uniform, which can be used throughout the world. Okay. Next, we'll be also talking about the seed sampling, in which we'll talk about the primary sample, we'll talk about the submitted sample, and then finally about the working samples. Okay, so these things we'll be talking in the seed sampling, plus like how the sampling is done. Lastly, like we will be talking in detail about the different TC testing methods. different seed testing methods, starting with, if I talk about the physical purity, if I talk about the genetic purity, if I talk about the germination, next we talk about the viability and the vigor and germination. Germination is done. Then seed health. Anything is left? Viability, vigor, seed health, germination. 4, 5, 6. Okay, fine. So these are the different tests that we are going to talk in this video. And one more thing. It is like moisture content. Seed moisture content. So these 7 tests we will be talking in detail. So what detail is necessary for you? That's most important. See, you need not to read each and everything, each and procedure of each and every test. That's not really required for the exams. So what is required from any of these things, like you have to make sure that you know about the submitted sample, you know about the working sample, and you know about the procedure plus the results that are interpreted. So these things becomes most important from the seed testing methods. Hope this thing is clear to everyone. Can we start? Okay. The first thing. Now, what is about seed testing? Like how this seed testing was initiated. See, during the 18th century or in the early 18th century or in the 17th century or early 18th century. So what happened is in the European country where the seeds were sold in the market. So Frederick Nobe, one of the pioneer in the seed testing field, who is also called as father of seed testing. So he came to know that, he noticed that for in one example, which was clover. So clover which were like white, red, these clovers were mixed with each other. So where he noticed it. Later, when fescue seeds were analyzed, he found that this fescue seeds were only 20% pure. The purity percentage of this fescue seeds was only 20%. So based on this, like analyzing these things, evaluating the things that is going in the seed market in the Europe, so he came to know about the importance of maintaining the quality. So that's how the seed testing was started. And the first seed testing laboratory that everyone should be well aware of, like which is the important point or the important question in any exams, which is like first seed testing laboratory was in and Saxony. Okay, so that you have to remember. It was in 1859. And the second seed testing laboratory, and can anyone tell me, so when was the second seed testing laboratory was established after this place? Second one, in which year? Quick, anyone? I need some answers from you. Like if you can answer, you will answer, then this session will be very much useful, and also like it will be very much enjoyable to everyone. Not seeing any comments till now. Anyone, like the second seed testing laboratory, like see, we have like complete seed testing to cover. It will take more than 3 to 4 hours if we go in this space. Second seed testing, it was in Denmark. It was in the year 1871. So what about third? It was in USA. It was in the year 1876. Okay, this is 1871 and this is in the year 1876. So this is about the word history of seed testing that you should be knowing about. Next, what is the connection with the ISTA? Before that, I'll tell you the purpose. So this purpose in India. In India, what is the importance of seed testing? Seed testing, there are the two important conditions when the seed testing is done. See, one is for seed certification. If you know about the seed certification already, then you can connect with this. The other thing is like for the seed quality control. Both are for seed quality control only, but this one, it is in the market already, where the seeds are sold in the market. Seed quality control for the seeds sold in the market. So how it is, for example, when it comes to seed certification, whatever seeds that are sold as a certified seeds, it has to undergo seed testing. So who is going to take care of all these things? It is by seed certification officer, the complete agency, right? So in this we go for seed testing, where the routine methods will be followed or the routine test will be done for the seeds to evaluate the quality. Once the qualities are met and according to the standards of IMSCS, it will be checked in the IMSCS. So the seed certification will be given. Similarly, the seeds which are sold in the market, which are like certified, which may be certified or non-certified. So this for this, we have a special provision in the Seed Control Order 1983, where the seed inspectors are appointed. So they look after the quality of the seeds in the market. Whenever they get some doubt, they will just collect the seed samples and they give it to the seed analyst, or they submit to the seed testing laboratories, where they get the satisfactory results. In India, so you have to remember that it is for mainly two different conditions. One, for the seeds which are sold in the market, and for the seeds which are going for certification. For certification, this is under the Seed Act, the complete provisions. These are under the Seed Act 1966, which is like most important thing that you have to remember. Okay. Similarly, this comes under the Seed Control Order 1983. But what about the steps, or what about the process of seed testing? See, these two are different things I told you, but what about the process? Do they follow the same process, or do they follow the different process? So is there any ambiguity between these two, or both are same? So this was actually a problem which was clarified by forming the one international institutes, which is ISTA. See, this was just an example. The ambiguity which I mentioned, it was just an example, not actually the reason. Okay, example where the different agencies or different seed testing laboratories or different countries follow the different process for the seed testing. So to bring the uniformity in the seed testing, what there were total of 4 seed testing congress. Yeah, so there were total of 4 seed testing congress which were held in the different years and in the different places. You have to remember all of them. See, all of the first 4 seed testing congress is very much important. So after that, in the 4th seed testing congress, the ISTA was established. In the 4th seed testing congress, which was held in the Cambridge, finally the ISTA was established, which was in 1924. The complete list is being given here. You can take a screenshot, and I'll be also sharing the PDF of this entire document that we have made, if it is useful for your exams. So just fill the Google form that is available in the description. Okay. So this is last one was UK Cambridge. Fine. This was in the 1924 when the ISTA was established. What is the full form of ISTA? It is International Seed Testing Association. I won't be writing it. You can understand, like, it is International Seed Testing Association. Why? What was the purpose of making it? It was mainly to bring the uniformity in the seed testing process or seed testing procedures. So this was the main motto of bringing the ISTA. Okay. Now, what is important for you, like, when was it was established? 1924, and it was in the 4th Congress. This is like seed testing congress. Next, what is important? The headquarters. Where the headquarters is located? It is in Zurich, Switzerland. This you have to remember. The most important one. Next, other things where like ISTA is India. Talking about the India. So India is the member of the ISTA. Okay, and in India we have like total of 8 ISTA accredited labs. So what are these accredited again? See, you can understand this in a simple words. We have like ICAR accreditation. So what exactly is ICAR accreditation? The ICAR will give a recognition or a badge, something like badge, to state agriculture universities or the institutes in the agriculture who meet the standards. Similarly, ISTA also provides that accreditation. So in India, we have total of 8 ISTA accredited labs. So out of that 8, 6 are private and 2 are government. Okay, in 6, like if I have to list, like it's Namdari seeds, you have Kala seeds, you have East and West, and you have Mayako, and you have like Indo-American. Okay, and we have like Nuzivedu seeds. So these are 6. Next, if I talk about the 2, one is Telangana State Seed Testing Laboratory. First, Telangana State Seed Testing Laboratory, and the second one is Coimbatore, which is located in the Coimbatore. The seed testing laboratory located in Coimbatore, which is Tamil Nadu. So this you have to remember. These 2 are government, and the 6 which I mentioned are the private, which are currently existing. Okay, and next, talking about the ISTA. So ISTA has total of 20 technical committees. And the accreditation process, like ICAR like goes for the inspection for the accreditation, right? So similarly, here the accreditation is valid for 3 years. Okay, that you can remember. Next thing, so what else the ISTA provides? The ISTA provides the certificates. So ISTA accredited labs in India, I mentioned there are a total of 8. Okay. Next, what are ISTA certificates? See, this is also most important. ISTA provides the certificates. The name of the certificates is seed sample certificate, and second one is seed lot certificate. So what is the difference? See that there is a major problem in understanding the difference. Okay, a lot of students confuse. See, this seed sample certificate, it is also called as blue certificate. You have to remember this. This can be a direct question in the exam. And the seed lot certificates, it is like orange certificate. How to remember these colors? O for O and O for lot. That's it. Wherever there is O, you can remember it is orange. Other one is like blue. It's a simple step, like how you can remember it. No confusions. See, what is seed lot certificate? What does a seed lot means? Seed lot, it is a physically identifiable space where the seeds have been kept, right? So if the certificate or the analysis, if the seed testing is done for the entire lot by taking the samples from the entire lot, then the certificate which is given is seed lot. And if the seed lot certificate is available, that means it is given to the entire lot. But if it is seed sample certificate, only it is provided to the sample that is provided for the testing. So only that sample is being checked by the ISTA, and the certificate is only belonging to that particular sample. So this is the simple difference between the sample certificate and the seed lot. You might also go for like one more certificate which is called as green certificate. Currently it is not in working, like you need not to worry about it. So this is somewhat confusing, so completely ignore it. That's it. Okay, write clearly. So you want me to write clearly and slowly? Is it so, or like you can understand like what I'm saying? Like others, like if you have any problem with the writing, please let me know. Like I'll just go little bit slower. Speed is okay, but I have to write it clearly, or you are able to understand like what I'm saying, pronouncing, or should I write it clearly? Because anyhow, like I'll be providing you the PDF. PDF will be provided to you. You can take a printout of it also. That's why, to save the time, I'm not writing the things clearly, but I'll try to write it. Okay, so this is about the seeds ISTA certificate. So if I have to again summarize like what I have said. So I mentioned about two things. One is seed sample certificate, and the second one is seed lot certificate. And this is called as orange certificate color, and it is like blue certificate. Okay, that you have to remember, and I will also explain to you like what are. Fine. The next, the Indian seed history. So coming to the Indian seed history, like what you have to remember and what is the exact history. See, in India, the seed testing works first started during the 1960s. Okay, the first seed testing laboratory that was established, as you all know, it is in the CSTL. Okay, so this was like Central Seed Testing Laboratory, and the pioneer members or the pioneer scientists or the pioneer of the Indian seed testing history, who are they? One is Douglas and Chellam and Amir Singh. So Amir Singh is also considered as like father of Indian or father of seed technology in India. But this is not pronounced in every textbook that you see, but somewhere you might find this. Okay, just know it as a student of like seed technology. That's it. So why they brought up with a manual, first Indian seed testing manual. Seed testing manual was first published by these three scientists. This is the contribution of this scientist, main contribution. And CSTL, one thing you have to remember here, it was about the funding. Who funded this? Funding for this CSTL was provided by Rockefeller Foundation. That you have to remember. That is very much important. And CSTL, where it was, see, in this also there is some confusion. Whenever we see the CSTL, there are two locations that we come to know. One is New Delhi and other one is Varanasi. So remember, when it was established, it was in New Delhi, but currently it is in Varanasi. So now, again, there is one more confusion with the students. It's about like CSTL and NSRTC. So in 2005, NSRTC was established. What is this NSRTC? National Seed Research and Training Center. Okay, and where it is located? It is located in Varanasi. And CSTL, where it is located? It is also currently located in Varanasi. But again, there is some confusion between these two, whether both are the same or it is different, and where it is located. Location wise, it is located exactly in the same place, but it is a part, like CSTL is a part of NSRTC. That's it. Okay, and this from 1960, it was shifted to Varanasi in the year 2007. These things can be asked in the exams. To Varanasi was in the year 2007, the CSTL. Currently, next important thing. Currently, how many seed testing laboratories are there in India? There are 2 government central seed testing labs. There are 2 government central seed testing lab in India. Where are they? Can anyone just comment in the chat box? Why people are not answering any questions? Am I asking like very difficult questions, or am I asking like very easy questions that you don't want to answer? Yes, anyone? The 2 CSLs, government seed testing laboratories in India. One is CSL, as already mentioned. It is located in. One CSL is located in. One is CICR Nagpur, and it is only for the cotton. That is hybrid cotton. Okay. The other one you have to drop it in the comment section. So one, yes, very good. So one has been mentioned like cotton research, but what about the other one? You have to let me know in the comment section. So this is about the Indian seed history that you should be knowing about. Okay. Next thing, seed testing. So these questions we'll take it later. We can discuss this question.
With this background, we'll go into questions now. So the question is, seed testing is performed to. The options are to determine the viability, or to determine the seed-borne diseases, and to know what is the healthy seedling status of the seed, and all of the above. So you tell me, what are the?
So no one is going to reply.
Be interactive. This session is for you only. After a thorough preparation, we did it we thought of doing this in one shot, where nowhere you will find this video, right? Won't get an entire seed testing in one part. So be interactive. It will be useful for you only. So as sir discussed, in seed testing, we are having so many tests where we are going to identify the quality of a seed. So quality of a seed means where we are getting moisture, germination, vigor, viability, health, right? So the answer is all of the above. So where we can determine the viability, where we can determine the healthy status, and also the seed-borne diseases, right?
And like I have to mention here about the blue book. So in the Indian seed testing, I was mentioning about the different things, right? See, I told about the 2 government CSTLs, and I told you about the Varanasi. So one more thing about the pioneers. I was talking about the pioneers in the Indian seed testing history. So here one more thing that should be mentioned, apart from the 3 scientists which I told, it is about the publishing of IMS. So IMSCS, which is also more commonly called as Blue Book, which is like Indian Minimum Seed Certification Standards. It was published by Conver and Singh. Okay, so you have to remember this. That is most important. It is in 1988. Okay. Same question has been asked here. So it is not like 1977. It is 1988. I think it is 1988. Yeah, it is 1988 only. So this is 1988. Okay. So blue book is written by this one. Next, seed sampling. So when we come to seed sampling, see when exactly we do the seed sampling. See, I told you the 2 important things in India. So when we do it, one is like when we are going for certification, right? And second thing, when the seeds are already sold in the market. In both the cases, if there are seeds that are being sold, or if the seeds that are kept in storage, how do we sample it? So sampling is a first step for the seed testing. See, one is seed sampling. Sample the seeds. Sampling means what? Seed sampling means like taking the seeds for seed testing. Second step is seed testing, which is like more complex, right? Before that, what do? We do a simple process called as seed sampling. What exactly it is? Seed sampling is a procedure where we will be taking the seeds from the lot, And this seed sample, whatever we are taking, they are called as primary samples. And this primary samples is combined to form a composite sample. From this composite sample, we give a certain amount of seeds to the seed testing lab, which is like submitted sample, from which there is again some sample which is derived for testing that for the different seed testing for the different seed testing procedures like viability, vigor, germination. That is called as like working sample. Okay, so this is the history about the brief intro about the seed sampling, where you have a seed lot. From the seed lots, you take the primary samples, and from the primary samples, you mix all the primary samples to form the composite sample, and then you go for the submitted sample, which is submitted to seed testing lab. Okay, and from this, the seed analyst will take the working sample. Okay, sampling equipment.
So as sir said, to sampling means since we can't do testing for all seed lot, which is really impossible thing, so we will take some sample which is a true representative of that seed lot, right? How do we take sampling? So we have some equipments called as their stick or sleeve type triers, knobhead trier, bin sampler, and sampling by hand also. Where we are using these different equipments, I'll explain here. So you can see this, the image is called a stick or sleeve type tray because it is having a two tubes, the inner and outer one like a sleeve. So it is by name you can see that, and with some pores where you can use it horizontally or vertically, right? When you have to take a samples from a very depth, you have to use it vertically, or like shallow depth, then you can go for horizontal use. So it is also called as bag triers because we are using it in a bag. Some containers also we will use it, right? So this thick or sleeve type trier is the most commonly used one, and here you can see this knobhead trier, which is a very long, like almost it is 1500 mm long, having holding here and short pointed tip here. We just keep this in a bag or in a container, and we will just rotate it 180 degrees so that whatever the pores are there in that trial, it will get open and seeds will be entered into that tube, and we have to close there itself and we have to take it out, right? That is the procedure of usage of knobhead trier. And the next one is bin sampler, where we are having a lot of like any bulk quantity or any we have kept our seeds in bins also, like in bags and containers. Then we go for bin sampler. So you can use, just we have to keep this in a bin directly. We have to rotate it. We will get the seeds, right? Like this. You just observe the diagram, and then hand sampling, where when we go for hand sampling, though it is very difficult method since we can't go for it, but it is recommended. Why? For chaffy seeds, right? To use all these samplers, the seed should be free flowing. Then only it will enter and we can close it and we can get it, right? But when we have a chaffy seeds, they're non-free flowing, right? They won't flow freely. Then we have to go for hand sampling. Just as depicted in a diagram, you have to just, with hand, you have to take a seeds. The very important note here is you have to close your hands very carefully so that you won't spill the seeds. Okay, then we have to take out our hands. That's it. That is the hand sampling. So based on this, we have a question here.
So while sampling from the seed lot of the 50 bags is taken, what should be the minimum number of primary sample? The minimum number of primary sample that should be taken. Okay, see what exactly is this? So there's a lot of confusion with respect to the seed sampling. A lot of students also ask me regularly regarding this, regarding the table that has been given differently in the different books. Let me clarify this again once. Okay, when we talk about the seed lots, there is a seed lot. Seed lots can be heaped into bags, right? It can be like bags of any kgs that are heaped together, which are stored, from where we have to take the samples, which we call it as primary sample. But how many primary samples should be taken, that entirely depends on the quantity of the seeds. It's like common sense, right? It depends on the quantity. If the quantity of the seeds is more, like, then we go for the higher number of primary samples. But again, there is a limit to that also. But what if like the seeds are kept in bulk? It is in the bulk storage. So we also have to go for primaries, taking the primary sample. But again, even though it depends on the quantity, but there is a difference between when the seeds are heaped or when the seeds are stored in the bags or the container, and when the seeds are stored in the bulk. The word is bulk here. Okay, and here it is like bags or containers. These are the two important terms that you should be knowing. From this, how many primary samples we take, that entirely depends on the quantity. So how to know this? So this is by following a table that is given by whom? That is the most important thing. See, if you look at the tables that are given in RL Agarwal and the ISTA, and in any other books, any other one-liners or any other objectives books that you refer, there is a difference. So ultimately, it is from the ISTA that you have to follow. It has been directly given for the seed lots or the seeds that are stored in bags and the seeds which are stored in containers and the seeds which are stored in bulk. So you have to directly follow the table which is being mentioned in the ISTA. No need to follow any other books. Okay. And there is one more confusion where even if you see the seed control order 1983, the document, even in this you might find one table which tells us about how to go for seed sampling. But do not even follow this. This is outdated now. Just follow the ISTA table where like things have been updated, and currently what we have to follow for taking the primary samples is been clearly prescribed in the ISTA. Right? So this is what you have to remember. Next thing, see, whenever you are going for sampling of 50 bags, so here the question is, I guess, like incomplete, because in each of the bags they have not given like what is the kg. The actual question should be, there are 50 bags, each containing 5 kg. Next, the question should be like number of primary samples should be taken. So this is a one standard question you might get. I'll ask other question. Same 50 bags, each containing around 26 kg, number of primary samples. Next thing, same 50 bags, each containing around 110 kg, number of primary samples to be taken. Okay. And fourth one, again here, so for this as a, okay, three conditions are fine to understand like what exactly I want to say. So what do you think the primary samples that should be taken in all the three conditions are same or different? Tell me, like who knows it? Anyone? The seed samples that should be taken for when the each bag is containing 5 kg, bag is containing 5 kg, each bag is containing 26 kg, and each bag is containing 110 kg. The sampling procedure or the table that we follow, it is same or different? Okay, different. I hope like you understood it. So there are like three different cases. Total cases are three. So what are the three different cases that you have to remember is, first case, if the seed size is less than 15 kg, there is a separate process. If the seed size is per bag or per container, it is like 15 to 100 kg, there is a separate procedure to be followed or separate table to be followed. If it is more than 100 kg, there is a separate table. So that is what you have to remember. When it is less than 15 kg, we have separate method, like different method to follow. And when it is between like 15 to 100, there is a different method to follow. And when it is more than 100, there is a different method to follow. Okay. So based on this, if you look at the table, different situations I told you. Again, if I have to compile, the different situations are, so one is if the number of seeds, like kg of seeds is less than 15. So second one is 15 to 100. And the third one is like more than 100. So these are the three different situations we have. In these three different situations, we have to follow the different methods in order to get the primary samples. What are the methods? First thing, if the seed lot is less than 15 kg, this is the one good example. Take the example here. There are total of 100 containers with 5 kg each. So what is the number of primary samples should be taken? So here the table that everyone knows. So this is the table that everyone knows, but this table according to the ISTA manual, so this is directly taken from the ISTA manual, so you can go refer it. This table is only applicable for the seed lot which contains like 15 to 100 kg. It has been clearly mentioned in the things here. Okay, it can be container or bag, don't get confused here. It might be container or bag, but it should be between the 15 to 100 kg. Then we have to directly follow this. But in the previous case, the example which I told you here, the number of seeds, like number of kgs of seeds is, or the kg of seed is less than 15, which is like 5 kg here. But total number of containers is 100, but in individually it is like 5 kg. So what we have to do? So in this case, we have to make sure that total this 120 containers into one sampling unit. Why 20 containers into one sampling unit? So this should go into 100 kg total units. So then we make it as like 20 containers is considered as one sampling unit. So there are total of 5 sampling units here, since 20 into 5 is 100. Total there were 100 containers, in which if I consider there are 20 containers. 20 containers into 5. In each container we have 5 kg. So total, when the quantity of seeds become 100, total quantity I'm saying in terms of kg. If you don't understand this, they are saying that 100 containers are there and there are total of 5 kgs in each. So that means in one container 5 kg is there. Okay, so this can be clubbed into 100 kg. When we club all the seed containers and make it 100 kg, that can be considered as one sampling unit, or this can be considered as one container as per the ISTA rules. In order to make that, how many containers should be combined to make it 100 kg? How many containers should be combined to make it 100 kg? Each container has 5 kg. That means 20 containers has to be combined, because 20 into 5 is like 100 kg. So this can be called as one container. Okay. So now how many one container, or don't get confused, it is called as one sampling unit. So now how many sampling units are formed here? It is 5 sampling. So based on this sampling units is now considered as container, and now we have to follow this table where it is 5 to 8, where 2 primary samples should be taken from each container, and total container that we are getting is 5 into 2 into 5, it is like 10 primary samples should be taken. This is what you have to understand. Okay, so this things was not clear earlier, so make sure that you understand right now. Even if there is some confusion here, let me know, like I'll just explain it again. See, the simple formula or simple rule to apply here is, if the number of the kg of seeds is less than 15, 5 is definitely less than 15, then we have to combine the different containers to make it at least 100 kg, and this 100 kg is considered one sampling unit that can be directly looked into the table. So this was the first condition. Second one, so this was the three condition which I mentioned. It is for what kind of seeds? These are for untreated seeds. Okay, these are for untreated seeds. If I talk about the treated seeds, with respect to the ISTA, for the treated seeds, there are different names for different formulations. One is seed pellets, one is seed tapes and TAPS. Okay, and another one is seed mats. For this, if the seeds has undergone some treatment, if it has gone pelleting, coating, if it has gone like made into seed tapes or seed mats, then what procedure to follow? If that is the case, as per the ISTA, what it says is, so for the seed pellets, granules, seed tapes and seed mats, the containers, if the containers is having less than 3 lakh seeds, this 3 lakh seeds, then again it should be combined to form a sampling unit not exceeding how much it is? It is 20 lakh. Okay, it has to be combined to form 20 lakh, and then from this again 2.1 table, this table should be again seen as a reference, and from this we can derive the primary sample. This condition, if by any chance if it is asked in the exams, so you have to know that if the sample is less than 3 lakh, number of seats I'm saying here, it is not in kg, the kg is not mentioned. 3 lakh seats. If it is less than 3 lakh seats, then you have to go for combining to form like 20 lakh seats, and that is considered as one, and this is considered as one sampling unit. And depending on how many sampling unit we have to look at the table. For example, if you have they give, there are 5 containers, each having 1 lakh seats or 1 lakh seats. So 5 containers each having 1 lakh seats, number of primary samples to be derived. And they also mention that these seeds are treated either let's say it is pelleted seeds. So this is for pelleted seeds. So this is the question that you get. So now if this is the question, as you can see, this is like 1 lakh seeds. This is definitely less than what is prescribed by the ISTA, which is 3 lakhs. So now in this case, you have to multiply 5 with 1. 5 with 1 lakh, it is like obviously like 5 lakh. So this 5 lakh is again less than 20 lakh seats. So the entire thing will be only considered as one sampling unit. So if there is one sampling unit, what you have to see, you have to just see this thing 1 to 4, and you have to take the 3 primary sample from the total thing. The total sampling unit is only 1. So total number of primary sample will be 3 primary sample here, because entirely it is considered as one sampling unit. So this is the other conditions for the pelleted seeds. Next, same thing, I have given the other examples now. Okay, so let me solve it also. Like if the seed lot is having like 20 containers and each having like 2,50,000 pelleted seeds in each, how many primary samples should be taken in the seed testing? See, if the seed lot is having 20 containers having like 40,000 or 4 lakh pelleted seeds in each, how many primary samples should be taken? So these are the two different examples. So in this is definitely less than 3 lakhs, which I mentioned. So what we have to do, we have to club it with the 20. So we have to multiply and make it as whole. 20 into 2.5 lakh. Okay, how much it is? It is 50 lakhs now. So this 50 lakhs can be divided into how many primary samples? So it has mentioned that 20 lakhs should be in one sampling unit. So first 20 lakh will be one primary sample, second 20 lakh will be second sampling unit, and the rest 10 lakh will be another sampling unit, third sampling unit, third. So first, second and third. So total how many sampling unit are there? Total of 3. So as per this, we have to just check the table. 3 primary samples from each container. That means each sampling unit. So in this case, the answer would be 3 into 3. It is like total of 9 primary samples should be derived in this above condition. So this was the first condition. Second condition, here it is mentioned like 4 lakh seats. So this is definitely above the bare minimum which is required. So directly we have to look at the table. 20 containers, so 20 comes here. So you have to follow this one. 15 primary samples should be taken, each from the 15 different containers. That's it. Okay. This is about the second condition. We'll come to the third case. Third case, it is with respect to the bulk. If the number of seeds are more than 100 kg, remember this, don't get confused. If it is more than 100 kg, one condition, or if it is stored in bulk, for both the things, we have to follow this particular table. If I have to give the example, if there are of 20 containers, each having 120 kg seeds, number of primary samples. So in this case, do you directly see that there are 20 containers and do you directly check this table and answer? If you're doing it, it's wrong. Why? Because each container is having 120, it is definitely more than 100 kg, which is prescribed by the ISTA. So in this case, we have to first multiply 20 into 120 kg, which will be 2400, and from this is like now total 22,400 kg, which is in bulk. That we have to see here. It comes in 3,000. Okay, 24,000 here. This last line or in the last row, we have to see like how many samples should be taken. So this comes here. after bulking. Okay, so this is how you have to sample it. So it was 20 containers, right? 12 to the 24, it was 2400 kg for 2400. So this comes here, sorry, second one. So we have to follow this second row for the seeds which are stored in bulk, or if it is more than 100 kg. So this is entirely about the seed sampling that you should be knowing. Similarly, see the sampling intensity for the seeds stored in bulk. So this is very simple. If they have given 3000 already directly, so you have to directly see this table and you have to answer. That's it. This is about the seed sampling. Okay, next.
Okay, we got a sample here, right? We are submitting a submitted, like that is called submitted sample for testing. How to obtain working sample from that submitted sample, right? Sometimes what happens, if it is very precious seeds and costly seeds, they will directly submit that much quantity of seeds, which is directly the working sample, right? If they have a very less amount of seeds, or if the seeds are very precious or costly. And sometimes what happens, they are submitting some bulk amount, that is called submitted sample, which is more than the working sample. Since we can't do it for complete submitted sample, we have to obtain a working sample which is a homogeneous, right? What is the main purpose of dividing the seed is to make a sample homogeneous as much as possible, because if it is homogeneous, then only it will truly represent the lot and we can assess the true quality of a seed lot. So how we are dividing the seeds, we have a different methods. So here, as I said, the main objective is to get a representative and homogeneous seed sample. We have a mechanical dividing, random cups, modified halving method, then spoon and hand halving method. In mechanical dividing, why it is called mechanical? We are physically or mechanically dividing the seeds using some equipments. Those are called dividers. Remember this. We have a different type of dividers. Let me show you this. Yeah. Corner divider, gamete seed type divider, and third type is soil divider. This is called as conical divider, since the shape you can see here, and the principle is gravitational force. What you have to remember here is what is the principle behind every divider, on which principle the dividers are working. For this conical divider or Warner divider, it is called the principle behind it is gravitational force. The name is, since it is a scientist who has given this, 1960. Remember this. And the second type is gamete seed divider. It works on the principle centrifugal force. In both the conditions, you just see the hoppers where we put our seeds, it will directly divide this into two halves. Are we using this directly? Since if we got a large amount of submitted sample, we have to obtain working sample, so that maybe in one occasion we are getting some sample. It may be more than our working sample. Then again we have to repeat the process. We have to take this and again we are going to divide it, and again also, right? In majority of the cases, in second time itself, we are getting the prescribed amount of working sample. If not, we can go for third also. Okay, we are repeating the process till we are obtaining the required amount of working sample. And what happens in soil divider? It is also similar to conical divider, that is corner divider, right? But what happens here is, when we are using this for large seeds and not of non-chaffy seeds, okay? Because in soil divider we are having the same condition, but where we are having large plates, it's almost 16 to 18 plates we are having. With two plates, if we pour seeds here, it will just divide seeds into two parts. Okay, so majorly for large seeds and large amount also we are using it, because we are having a wider channels here. These are called 18 to 16 plates or channels. Okay, so it will also work on the same principle, that is gravitational force. Remember this. So here is a question for you, as just Now we discuss conical divider, or it is also called as Werner divider, works on the principle of. You have to answer this. Okay. So conical divider, what is the working principle behind the conical divider? Options are like this: gravitational force, centripetal force, centrifugal force. Right. It is gravitational force. And also tell me what is the principle behind gamete seed divider? Gamete seed divider. Just now I mentioned it is centrifugal force. Don't confuse with centripetal force. Okay. Right. See. And I also told there are different methods of dividing, right? This is mechanical, where we have 3 different kinds of dividers. Accordingly we use. And random cups method, what we are going to do in random cups, just name itself indicate we are using a cups method, right? Yeah, it's almost 6 to 8 cups we are keeping in a tray. Okay. So after preliminary mixing, usually we will mix the seeds. After preliminary mixing, we are going to pour the seeds into cups. Randomly we are pouring, okay, in all the cups. Usually in a tray, wherever we have kept the cups, we are just going to pour the seeds. Whatever the seeds they are poured into these cups, we are selecting those seeds, okay, randomly, so that this randomness make you homogeneous. That is the principle behind why we are going for random. Okay. So whatever the seeds we got in these cups, we are going to take those as a working sample, if that is the required amount or prescribed amount, or else we are going to repeat. What are the seeds we got in this, we will take that again, we repeat the things with the seeds whatever we got in a first occasion. Okay, that is random cup method. And modified halving method, let me tell you what is hand halving, so that you can connect with the modified halving method. In hand halving, the name itself indicates that hand halving with for chaffy seeds and for non-free flowing seeds, we are using this one, hand halving method. What we are going to do here, just see the image. Here we got a working submitted sample. Here we are going to divide it into 2, then again into 4, again into 8, right? So in 8, we are going to retain 4 alternative and we are going to remove other 4. If we have a 4 halves here, okay, then we are going to retain alternative types, okay, yes, like this. And whatever the other 4 will be removed, and what these 4 will be mixed. If it is the more than working sample, again we are going to repeat the process. Again from 4 we are going to do it 2 to 4, 4 to 8. Again we are retaining the alternative one and we are taking the other 4. Okay. This is called as hand halving method. The same way, what happens in modified halving method is, we are using some equipment which is having a plates or bottoms, we call it as, each alternate bottom with no bottoms. Like, see, here it is having a bottom and it is not, and here it is having, it is not. Okay. So whenever after pre-mixing of the seeds or preliminary mixing of the seeds, you just pour into this one, and whatever the seeds poured into this, we are going to select this. Since because exactly half will be poured into the cups and half will be into the bottom, right? So just understand this. The modified halving is, we are using some equipment. In spoon method, the name itself indicates when we are going to use spoon method. So whenever there is a single small seeded species, right? You can't use some like conical dividers or soil type dividers for such a small seeded species. That's why we are going for a spoon seeded method. What we are going to do in spoon method is, in a tray, the even tray, just remember this. We are going to just pour the seeds, and with spatula in one hand and spoon in the other hand, we are just going to pick a seeds, at least 5 random places. Okay, just minimum 5 random places we have to select, and we are going to pick a seeds, whatever the seeds we got, right? we are using spoon and spatula. Whatever the seeds we got from this method, we are considered as a working sample. So accordingly, if it is for purity, that is different. If it is for germination, the working sample is different. So just remember this. This is spoon method. So based on that, we have question here. Hand halving method is restricted to chaffy seeds, option 1. And spoon method is suitable for samples of single small seeded species. And random cup is suitable for brassica species. And soil divider is suitable for large seeds and chaffy seeds. Tell me, what is the answer for it? It's a very important question which is already asked in some PG, PhD, SRF exam. So just tell me. So how many statements are right? You just tell me how many statements from here are right. Okay, let me explain you. As I told, hand halving is for chaffy seeds. Spoon method is for single. This is also right. And random cup is suitable for brassica species. In random cup, where we need is just remember this 10-gram working sample. We are going to do that. Okay. And such species which should not roll. If the seeds are rolling like a brassica species, how can you pour the seeds into cups, right? So this is wrong. For those which do not roll like brassica, so it is not for brassica species. And soil divide is suitable for large seeds and chaffy seeds. For chaffy seeds, we can't use a soil divider. Okay, for large seeds we can. So first and second are right and third and fourth statements are wrong. Okay, the question is here, the random cup method is suitable for sampling of how many gram seeds? It is very important since it is already the prescribed one. First, it is for 10-gram seeds.
So this completes the things which I mentioned. So from the seed testing, I told you from the starting what are important. First thing I mentioned, word history is important. That we talked about. Second thing, we talked about Indian seed history. Right. Then we talked about the, and we also talked about is certificates. Then talked about seed sampling. In seed sampling, we talked about two key areas. One was taking the primary samples, and the second was the method of seed sampling. Okay, now what is the next thing? What is the next thing of after sampling? What is the next thing after seed sampling? It is starting the test. So now, what are the different tests that we do? Like we have like majorly 7 different types of tests which are done for the different purpose. First one is called as physical purity test, which is done to know the physical purity of the seed lot. Second one is genetic purity test. Third one is germination test. And fourth one, we have seed viability. Fifth one, we have seed vigor. And sixth one, we have moisture content of the seeds. Last is, as I mentioned, seed health. Okay. So if we are able to, know about what is the working sample, for all the tests, plus what is the procedure of this test. So most of the things will be covered for the exam. So this is what exactly you have to remember. Procedure, or like reporting how the results are reported, and if there are any specific principles behind the test. These 3 things becomes the most important one from the seed testing part for the 7 different tests done for the different purpose. Okay, so from that, let's start from the first one. First one is the physical purity test. So what exactly is a physical purity test? Physical purity is nothing but just to evaluate the purity of the seeds physically, just by observing with the naked eyes. Like if I have a seeds of wheat, physically I have to only have, if I have to say like I have a seed lot which has a seeds of, let's say, wheat. So if I have to say that it is 100% pure, physically pure, that means all the seeds which are available, all the material which is available in this lot should be compulsorily wheat seeds. What if there is some stones here, What if there are pellets here? What if there are some inert matter? Neither they are seeds, neither they are something. So if these are mixed, I cannot call something to be like 100% pure, right? So in every seed lot, there will be certain extra additional fractions along with the pure seed. How much fraction does the really seeds have, the seed lot have? That can be calculated by knowing about the physical purity percentage. That is what we are going to discuss. So I talked about the different fractions. What are the probable fractions? See, according to the ISTA classification, ISTA has made into only 3 fractions for the physical purity. That you have to remember, which is very, very important. The first fraction is pure seed fraction. Second one is other crop seeds. Third one is inert matter. Okay, so this is from the ISTA classification that you have to know. But when it comes to the Indian classification of seed testing, in India, we assume not 3 but 4. So 3 are same, but the fourth one is like weed seeds. Why this is being included in India? Because the seeds which are considered as weed are different to different countries. So whatever seeds that are considered, or whatever crops that are considered as a weed, so that will be included in this, the fourth fraction, which is the wheat seeds. So now tell me, like if I say I have total of 100 seeds, I have counted the 100 seeds. So how now you will definitely ask, okay, someone is asking about like BSc horticulture. There is no batch for the BSc horticulture. Can I study from this JRF horticulture batch and will I understand it? Yes, you can definitely get it, because whatever we have done in the horticulture batch is from the scratch only. So if you are interested, you can definitely go for batch. So if you need any details, just let me know. Contact us on this number. So our team will definitely help. So coming back, see in this India, like we have 4 different fractions. So let me give you an example. If there are total of 100 seats, now the one very common question that will come. So if I ask you, what is the method that we follow to do the physical purity testing? Can you tell me? Someone asked like how to join the main class of ICAR JRF 2026. You have to just enroll the course. To enroll the course, just visit our website www.agriaddict.in. There you can find your course for your subject. You can enroll for it and directly you can take the classes. Yes, anyone? Can you just tell me like what is the standard procedure? For example, just example, very good example. Seed viability. If I say seed viability, you will definitely say tetraxylum test. Yes, we use the tetraxylum test to check the seed viability. So similarly, if I ask you, so what procedure we use to do the physical purity testing? So how it is done? What's your answer? Yes, anyone? I'm expecting some answers from you. Come on. So we were actually planning to cover all the 8 units of seed technology, which is useful for the SRF, which is for the ASRB, and it is also useful for all agriculture exams. But if you feel that we have to do it, then all about the interaction. The more you comment, the more you answer, only then we will be able to come up with that. Purity board, exactly. See, it is a simple table examination that we go for. I already told in several times in my class also, the PG class, PhD class, it is like table examination that we do in a purity board. Simple purity board that we keep, and we see if this is a purity board, in one side we'll have all seeds. So we'll just count and keep the sides into 3 different fractions. So pure seed we'll keep it in one different side, one different corner, and in inert matter in one different corner, and other crop seeds in one different corner. If we have to make the 3 fractions, if I have to make the 4 fractions, then I will make one more corner and I'll keep the other one, and I'll count the number of seeds. For example, if I count total 100 seeds, if I come to know that there are total of 98 seeds which are of that particular crop, and next the one seed was like inert matter, and one seed was like from the other crop seed, which is not entirely the one which I am checking for. So what is the exact purity percentage here? Can anyone just quickly? Yes, Saurika. Only Saurika is answering. Manisha is answering earlier. Okay. Farooq was answering good. Anyone? Okay. It is simply a 98%. So this is how much simple it is. Very simple to understand. It is 98% that you have to know. So from the exam point of view, these fractions, the ISTA fractions and the Indian fractions, that are important. So please make sure that you understand this thing. See, apart from that, what else is important? There are also few other things are important from the purity analysis. What is it exactly? Yes. So working sample, okay, I'll complete the working sample. So in the working sample, another very important thing that has to be noted for the purity analysis, the working sample is 2500 seeds, which should be less than or equal to 1000 grams. Okay, so this is the working samples that is to be taken or that is to be counted, like 2500 seeds less than or equal to the 1000 grams. Maximum is like 1000 grams. This is the working sample for the purity analysis. Now she will explain you about the other things which are to be actually noted and studied in detail for your PSG and the ASRB net exams. If for other exams, you can ignore it.
As sir said, we have a 3 components, right? Pure seed and inert matter and other crop seed. When we consider it as a pure seed, okay? So we consider it as a pure seed, or first of all, tell me when we consider it as a pure seed. Whatever the seeds which is stated by applicant or majority of the portion, if we have a wheat, we have a almost wheat proportion in 98% or 99%. Just here and there some rice seeds. Then we, without even saying it, we call it as a wheat seeds, right? And in that rice is other crop seed. We won't say rice is the crop or a main crop and wheat are the other crop seeds in that. Okay, the majority of the seeds, or if it is stated by an applicant, that is called pure seed. And one more, the basic one is more 50% rule. Okay, 50% rule means where more than half of the size is of a main crop or are considered one is under consideration, then also it is called as pure seed. If we have a seed which is broken one, but that is, though it is broken, it is more than half, it is, if it is present, then also we consider it as a pure seed. Just remember. And if we consider some capsules or any caryopsis or any pods, by saying itself, if we say, okay, if the seeds are present with just naked eye, without using any diphenoscope or anything, then also we consider it as a pure seed. Unless you see there is no intact seed inside, till that we consider it as a pure seed. By seeing itself, if you can say this pod having no seed at all, then only you can consider it as a inert matter or anything, any other component, right? So the 3 important one is it should be major one and it should have more than half. It is not equal to also, just remember it is more than, not more than or equal. This is important. And also the with intact seed. Okay. And here we are having different families in, since the nature of a seed or size, whatever that nature, with that we are having a different kind of seeds, and accordingly we are following the different rules also in determining whether it belongs to pure seed or weed or any other component. So in families like, just remember this, LCCPT. So Leguminosae, Cruciferaceae, Cupressae, Pinaceae and Taxodiaceae. Very important are Leguminosae and Cruciferaceae. In these families, if the seed coat is entirely removed, here the matter is about seed coat. Okay, we have to see seed coat here. Okay, in Brassicaceae and Leguminosae especially, if the seed coat is removed, then it is regarded as an inert matter. Though it is having a seed with all the essential structures, we can see the embryo part, everything. But if we don't have a seed coat, then it is regarded as an inert matter. Okay, and in case of Leguminosae, right, it is dicots. In dicots, the separated cotyledons are also regarded as inert matter, irrespective of if it is having a radical plumule axis or if it is more than half of the testa is also present, the seed is also present, everything is present. But if cotyledons are missing, in dicots, these are the essential structures, right? If cotyledons are missing in Leguminosae, it is termed as an inert matter. Okay, just remember the points. In case of Graminaceae, or it is Poaceae family, Remember some species like some genus Lolium, Festica, and Elytrica repens. Not even all the species also. Okay. In Lolium, Festica, and Elytrica repens, flow rate with the caryopsis 1/3 or more than of the length of the palea. So we are having spikelet, right? With different number of flow rates, lemma, palea, what we discussed in the previous class. It should be more than 1/3 of the palea. Okay. From base of the radula, when we measure it, if it is more than 1/3, then the flow rate is more than 1/3 of the palea, then it is regarded as a pure seed. If not, it is regarded as an inert matter. Okay, then the presence of caryopsis, that is seed, in a Poaceae family is not always obligatory. Like we said, unless there is no intact seed, if we are having a caryopsis with a flow rate, if it is more than 1/3 of the palea, we are considering it as a pure seed. Okay, so we are not seeing whether seed is present or not by opening that all the flow rates, all the outer and inner bracts and all here. If we have that caryopsis more than 1/3 of the length, then it is called as pure seed. And in some species, like just remember this, Pova pratensis, Dactylis glamerata, and one more Pova species is there. Okay, for all these three species, where uniform blowing method is compulsory. For other species, how we are using this purity boards, right? Just a table examination. But these three species, uniform blowing method is used, and one more, Chloris Guyana. Okay, remember this. We can use this is if necessary we can use, but it is not obligatory. But along with these three species, the uniform blowing method can be used in Chloris Guyana also. In that case, so the uniform blowing method is followed. In that, what happens in uniform blowing method, just let me explain you in short. We are just going to blow the seeds for just 2 minutes, exactly 2 minutes. So what happens? We are getting one heavy portion and lighter portion. Heavy portion is a pure seed component, right? And just look for the other crop seeds. If other crop seeds are present in pure seed or heavy portion, we have to remove those. And other whatever all the seed coat, testa and everything present in lighter portion regarded as a inert matter. Just that is the difference. Just remember heavy portion and lighter portion in case of uniform blowing method. And multiple seed units like achenes and caesocarps in case of carrots and all, if they are left intact in the pure seed fraction. So while removing, I told right, in lighter we are getting some testa and all the dust. So in case of this Dactylis and Festica, we are not removing these multiple seed units. Just remember that also. And in case of some Dactylis, Festica, Pova, Sorghum and Triticum spelta species, the attached sterile florets are also not removed. Some sterile florets are considered as inert matter since it is of no use, right? In some species, but some species we are not at all removing those and we are just considering them directly as a pure seed. Okay, for certain genera, I told right, these appendages are left on the seed, but when they are reported, if that appendages are constituting for more than 1%, we are just reporting those on analysis certificate, but they are not removed. Okay. So family, according to family, we have a different definitions for a pure seed. Like if it is different in Leguminaceae, it is different for Malvaceae family. For Cucurbitaceae, we have different. So we are just providing those notes. Don't worry. Just remember the exception cases in Graminae family and also some uniform blowing method, how we are using, where we are using, and what is the working sample for that also. And that is about pure seed component. And for other crop seeds, just I mentioned, whatever all the component other than pure seed and inert matter, so just other than that of the pure seed as a which is grown as a crops, that is other crop seed. In wheat seeds, we know already we are having bulblates or tubal, which are recognized as objectional weeds or noxious weeds by laws or any official regulations. They are whichever we considering as a weeds in our India, they are regarded as a weed seed component. And inert matter, they include some seed units, that is like appendages, like any removed lemma or removed palea, awns also sometimes, right? All these are regarded as a inert matter.
So based on like weight of working sample, if the weight of working sample, if it is less than 1, and then total number of decimal places which are needed to be kept, it is like 4. This is very simple to remember. This is for the working sample for the physical purity analysis. If it is like 1 to 9.9, then the 3 digits, like it is like 43210. Here it is less than 1, 1 to 9, 10 to 99, 100 to 999, and 1000 and more. So that is how we can remember. Just remember that less than 1, 1 to 10, or 1 to 9, then you can remember as like 10 to 99 and 100 to 999 and 1000 to more. So on the left side and the right side you can write it as 4321. That's how you can remember the decimal points. So here there is one problem with the physical purity. The things like what Gayithri has mentioned or Gayithri has explained, you might have not gone through this earlier. Okay, this is all from the and it is also available in the RL Agarwal, but things will definitely not be clear in the first reading. So if that depends on your target, like if you are wanting to crack this ASRB net exams, or if you are studying for the ICAR PhD exam in subject of seed science, then this becomes like one of the important things. See, any question can come from here, so you have to ensure that you have to go through it at least like 4 or 5 times to understand this thing. Okay, nothing is like complex, nothing is conceptual. That's the conditions you have to remember. Otherwise, like if you are not interested to go for this, you can definitely skip it. Okay. So anyhow, like whatever she has explained, if even if like things are not very clear, or if it is boring, then just go through the PDF. The PDF I'll be sharing it in a like description box. To get the PDF, just fill the Google form as I already told. If you have already done it in the previous case, in the previous videos, just open the Google Drive link. In that Google Drive link, you will find this particular PDF plus the one-shot video of seed testing PDF in that, which is freely available for everyone, which you can take a printout of it and use it for your exams. Okay. So here now, talking about the real value of seeds. What is the question says, like what will be the real value of seeds if the purity percentage of the seed is 99% and germination percent is 91%? So what we have to remember, I'll just tell you the few key points here. The first one is, if the purity percentage is more than 98%, it is more than 98%, then we consider that it has their quality of seeds is very good quality seeds. Or the one specific term that we use here is like superior quality seed, if the physical purity of any lot is more than 98%. So now what about germination test? If I ask you one question, okay, I have to do the germination test, so I have to take the samples. Whether I will take the sample from the submitted sample or from the pure seed fraction, what will be your answer? I will take the working sample for the germination test, the working sample, which is like 400 seeds in total. That is like 100 seeds in 4 replication. This working sample, will I take it from the submitted sample directly, or will I have to take it from the pure seed fractions? Can anyone tell me quickly? This is important. That's why I'm asking you people. Yes. Yes, Manisha, Saurika. Anyone? Farooq? Sherin? So here there is a condition that pure seed. See, if I have to do the germination percentage, before that I will be already doing the purity percentage. Physical purity percentage will be already known before I go for germination test, if I am a seed analyst. So in the first case, if the physical purity of the seed lot that I am doing is more than 98%, if it is more than 98%, then then I can take directly from the submitted sample if it is the physical purity is more than 98%. I have already tested. But if the physical purity is less than 98%, then I have to take the working sample directly from the pure seed fraction to estimate the germination. So here, what is the conclusion? What I am trying to say is this germination and the physical purity, they are like complementing the exactly complementing the seed quality. So since they are completely complementing the seed quality, the real value of the seed, it will be always the germination into purity percentage. If in the exam, if you are being asked with the real value of seed, that is equal to germination percentage into purity percentage divided by 100. As simple as it is. I hope now it is clear, like why I have told you all the background story. And in that background story, the condition of more than 98%, if the physical purity of the seed lot which is given, it is more than 98%, then for germination test, we can directly take it from the submitted sample. This is also very important. Also remember this. And for the physical purity, if it is less than 98%, then we have to take the working sample for the germination test from the pure seed fraction, PSF, pure seed fraction. And what is the reason? Like because for the seed quality, the physical purity and the seed germination complement each other. That's why also the real value of the seed is always the physical purity percentage into the germination percentage divided by 100. So remember this formula. This is very much important. Next thing. So physical purity standards in okra. So the another very common question that is being asked for seed testing. They will simply ask you the physical purity of any crop. They will ask you. They'll ask you genetic purity of any crop. They'll ask you the germination percentage of any crop. So these three things are like most common things that you have to know in case of seed testing, which you can't definitely skip. So you can't or you can never skip this part in case of seed testing. So physical purity in okra. Why have they have specifically asked in okra? Because the okra or bendi, this has the highest physical purity standards, which is 99%. That's the reason they have asked here. Most commonly they either ask the extremes, either the higher extremes or the lower extreme. For example, they have asked about the soybean. Now what would be the for the soybean? To understand this, you have to go for this step. So you can take a screenshot, otherwise screenshot is even not required, like I'll be anyhow sharing the PDF directly. So I have compiled all the things. See, if you are not sure about like what data I'm given, if you are still having some doubt on this, you can definitely go for IMSCS. As I told you in the starting, I'll also mention the source. It is IMSCS which India follows in order to certify the seeds. So you can directly go and compare it with the IMSCS. I have taken all the information from the IMSCS only. Okay. So now to simplify it, to understand it in a better way and to remember it, I have clubbed it. For all major crops, it is 98%. But when it comes to the millets, except for sorghum and bajra, which is like 98, all millets have like 97. Rapeseed and mustard, sesame, jute, and all other have included here. Then groundnut, asparagus, spinach, so and so, like it is 96%. And next, it is tobacco, amaranth, carrot, it is 95%. And the highest is for the bendi, is like 99%. Okay, that's what you can remember. And this soybean. So soybean, if it is not mentioned in any other table, it is definitely 98%. So answer for this question would be 98. Okay, not present. So let us see the table again. So here the soybean, yeah, it is for 98%, 98 only. Soybean, it is 98%. Even if you people ask me directly, sir, do you know the physical purity percent of all the crops? No, like when I have to give some exam, I'll definitely try to remember this table. I know about the highest and lowest, to be honest. So highest and lowest, bendi, I know it is highest. For all other crops, I know like for major crops or majority crops, it is 98. But for other things, you have to just revise it, remember, and try to revise it again and again so that you can remember the exact purity percentage. Okay, so that is for the standards for the certification. Okay, now there is one more term is like what is this dockage? Whether it is like purity percentage of the seed or it is an impurity percentage. See, this dockage is actually the impurity percentage of seed. The term dockage, what does it mean? It is the impurity percentage that is present in the seed lot. So that is what dockage means. Okay. Next, there is one more question. So which among the following is correct? Generally, 2,500 seeds are used for the physical purity analysis. Okay, pure seed, other crop seeds, and inert matter are the components of purity analysis. Seed quality is superior if the pure seed percent is more than 98, and the gamete seed divider works on the principle of gravitational force. So now, quickly answer. Anyone? Sarika, is it for previous one or this one? It is for previous one. Just try to answer this question quickly. Anyone? Yes, anyone answer this question? Like, this is very simple question. Okay. The first thing, the generally 2005 we already discussed. This is true, this is true, this is true. And the last one, the gamete seed divider, whether it is working on gravitational force? No, it is from the centrifugal force. And which divider works on the gravitational force? It is like Bornier divider, or it is also called as conical divider. Fine. So based on this, answer would be like 1, 2, and 3. Okay, good. You are answering. Next, a seed lot with the 98% purity means that the 98% of seed will germinate, 98% of seeds are free from disease pathogens, 98% of the seeds are having the eye moisture content. So this is somewhat confusing. Try to decode it. If I say that 98% purity, does it means that there is a germination of 98%? If this was true, then the physical purity percentage should be equal to germination percentage. Is it true, this thing? So whatever purity that we have, that much germination we'll get. Is it true or no? Yes or tell me yes or no. Quick. No. Okay. So second thing, the 98% of the seeds are free from disease pathogens. Is it true? Third one, like 98% of the seeds have like eye moisture content. See, this purity percentage, let me tell you, like whatever we are trying to find from this, we are only finding how many pure seed we have. We do not get any information of whether they germinate or not, whether they are free from diseases or not, how much percentage of moisture they have. None of this is are true. Here, like 98% purity simply means that the pure seed fraction is equal to 98%. Okay, that is what it means. You have to understand the concept. Do not get confused. Okay, next thing, germination test. So in the germination test, again, we have different methods. So Gayatri will explain about all the different methods of germination test. But what I will tell you, I already told you about the working sample. The working sample is, it is like simply 100 seeds in for like 4 replication, which comes up to the 400 seeds. That's the very first thing that you have to remember as soon as you see the germination test. And what other things, what are the conditions? Simply like germination test, where it is connected, it is definitely connected in the labs. Okay, and the duration of this test, that differs from crop to crop. It initially like 2 to 3 days, till it can go for like 30 days also. That depends on the crop, like how much time or how much time does the crop generally take for germination. This is about the duration. And what about the conditions, other conditions? What about the light? What kind of light is required? It is like white fluorescent light is required for the germination. And what about the temperature? The ambient temperature of 20 to 25 degrees Celsius should be given in the lab. And if I talk about the relative humidity, it should be like mostly more than 90%. So this is the preferable condition for the seed germination test that will be conducted in the lab. And talking about the germination test, like types, we'll explain about the types of germination.
So with this previous slide, we can come to know that we have to provide light, temperature, relative humidity, and proper moisture also, right? So just remember, germination test means it is conducted under optimal conditions. Okay, this is a very important question. You can't go for field conditions directly. So in a controlled, that is optimal conditions only, we can conduct the germination test. What else we need other than light, temperature and all? We have substrates, right? We have different types, that is top of the paper method, between paper method, and sometimes we are going to use sand also, sometimes soil, and sometimes we are going to use some organic media also. But majorly we are going to use top of the paper method and between paper method, and other paper we also call it as a pleated paper method. Just remember this. These are the different types of substrate or type of methods we are going to use for germination test. So if you see here, in paper substrate we are going to use top of the paper, and that means we are going to use Petri plates here. In Petri plates we are going to keep seeds in a filter paper. In between paper we are called as germination papers by checking the phytotoxicity of paper. Okay, because if we want to use any substrate, it should be non-toxic and it should be porous, so that the moisture should retain also, and moisture should end will it should give water to the seeds for germinating, right? So just remember what are the basic conditions if you want to choose. If you want to choose any substrate for germination test, what are all the minimum basic requirement it should meet? Okay, and sometimes we are going to use sand, right? Like in sand, if we are going to use top of the sand method also. In so before that we are going to sterile it, and we are not reusing this. Remember, you should not reuse this also. Sometimes organic growing medium like vermiculites also we are using. Okay, so top of the paper method means we are placing the seeds one or more layers of the paper, and we are just allowing to germinate by keeping it in a germination chamber. Remember this. The same way, in between paper method also, seeds are placed with the two horizontal layers, and we are just keeping the seeds on one layer, and we are covering it with the other layer. We are just rolling it. That is called roll towel paper method, or we are just making a pleats from that paper, and then we are keeping the seeds in a uniform spacing, and then we are just wrapping it with a wax coated paper above, just to retain a moisture, right? This wax coated will prevent a moisture loss. Then we are keeping it in a germination chamber, and after two different crops the duration of the test will vary. Then we are going to take a counts. So once we have completed germination test, for example I am saying for wheat I have kept for between paper method, and I just kept for 4 to 8 days now. So just remember, for wheat it is 4 and 8, that is first count and second count. At the final count I want to assess the germination percentage of that seeds. Then there are 6 categories. Okay, for every test you have to remember what are all the components, what is the working sample, what is the submitted sample, and how we are interpreting the results. That means if it is in fractions, or how many fractions, till how many decimal points we are using it. Okay, just remember all these basics. So for this we are having a 6 categories here, like normal seedlings. When we call it as a normal seedlings, if we by just seeing this we should know, we should just assess like okay, this seed can able to germinate very well and it will grow into a very good plant in the future, right? So just to see that if we having a seed with good shoot, root, and what are the essential structures we call it. Sometimes it is a some lateral roots in case of uh, Poaceae family, tap roots or main roots in case of dicots, right? Some hypocotyles in monocots, epicotyles also. So what are all the essential structures? If we see it very clearly, then we call it as a normal seedlings. Sometimes there are slight defects, so just some slight defects, then also we are considering as a normal seedlings. So in normal seedlings with intact seedlings, there are 3 categories: intact seedlings, slight defects, and with the secondary infection. That means the infection is not from the mother seed. That may be from other factors. Maybe the environmental, like what are the surrounding environment also. That is secondary infection. Then we are considering as a normal seedlings. If it is abnormal seedling, that means that does not have all the essential structures. Just remember, you have all the shoots and everything. If it don't have a root, how can you consider it as a normal seedling? Right? It is completely damaged, deformed, or decayed. Just remember these three terms: DQ, damaged, deformed, or decayed. Then they are considered as a abnormal seedlings. And the third category is dead seeds. That means they're neither hot, neither not dormant, and they have not produced any part of the seedlings. By seeing itself, you just say that the seeds are dead. That is also other type of category, that is dead seeds. And fourth is hard seeds, mainly where we see it in Leguminaceae family, right? Why it is called hard? The seed coat is very hard in such a way that it will prevent the absorption of the water. It won't even absorb the water or imbibe the water. That is why it is called as hard seeds. And dormant seeds. So viable seeds other than hard seeds which fail to germinate when provided the prescribed germination conditions. So you know the difference between dormancy and quiescent phase, right? In quiescent means there is unfavorable conditions, that is why seeds is not germinating. But in case of dormant, though it is having all the favorable conditions, it is an adaptive mechanism that the seeds won't germinate even though you have provided all the light, moisture, what are all the conditions it needs. So that is called as dormant seeds. That is the other type of category. And fresh and ungerminated seeds, FUGs, we call it as. So seeds which have imbibed the moisture, that means they have already absorbed the water but they have failed to germinate, or it may be dormant also, right? Sometimes. So they are called as fresh seed. So remember 6 categories. Just remember, this is also very, very, very important because all questions will ask around this topic only. There are two different types of germination, right? That is hypogeal and epigeal, where we have other hypoepigeal also, some Papaver species. Just keep it apart. Just mainly I'm focusing on two types, that is epigeal and hypogeal. The name itself, it indicates epi means above, hypo means below. So the difference is about cotyledons. If the cotyledons are below the ground, if they remain below the ground, that is hypogeal. If cotyledons will rise above the soil or ground, that is epigeal conditions. You just see here. This is a soil level or ground level from where this hypocotyle is elongating and cotyledons, the leaf-like structures, right? These cotyledons will come out of the ground. That is epigeal. Here, in case of only epicotyle is elongated and it has raised above the ground, but these cotyledons, you just see here, the cotyledons and hypocotyle, everything is remaining below the ground. That is hypogeal. How to remember this? Okay, we have given the short, you know, in case of monocots and dicots, most of the monocots, okay, most of the monocots are hypogeal germination, but the exceptions are allium species like onion, garlic, okay, and most of the dicots are epigeal germination. Here I want to say, especially in case of pulses, okay, we have Rabi pulses and Kharif pulses, right? So in case of pulses, all Kharif pulses will show epigeal germination except red grass. Since there are so many things to remember in seed technology, you have to focus on those exceptional points so that you can remember very easily, right? You can't remember all the pulses which is epigeal, which is hypogeal. So just remember what are all the Kharif pulses are there. They will show epigeal except red gram. And what are all the Rabi pulses are there? They show hypogeal except rajma. Okay. And other than that, all other monocots like soybean, brassica, castor, cucumber, everything, every crop will show apigeal germination. Okay. So the question is here, during the germination test, following seedlings are considered as a normal seedlings. Okay. The options are intact seedlings, decayed and deformed seedlings, seedlings with slight defects, seedlings with a secondary infection. Choose the incorrect one. You have to read question carefully. So choose the incorrect one. Answer this. But still people see. This is really interesting, right? It is a midnight 12 o'clock we are teaching and you still here on a live session. That's how the things will work for exam preparation. Really so happy to see you all. Still, even though it's 12 o'clock, you guys are so interested and dedicated to listen to this session. So just try to answer this. Be interactive. I know it's already almost 1.5 hour session, which is really boring. But the things are like that in seed testing. Seed testing in one shot means you have to cover each and everything, where the majority 7 tests are still we are in germination test. We'll going to complete it very soon. Okay. Right. People are answering it right. So the incorrect one is only B. That is decayed, deformed, and decayed, damaged, and deformed. These are considered as abnormal seedlings, and all other with intact, very slight defect, if it is having secondary infection also, they are considered as a normal seedlings. And the next question is, as sir said, the majority of a question is about factual things, right? In seed testing, because we have so many facts, we have so many exceptions also. That's why they may ask, what is the germination percentage of this crop? What is the physical purity of that crop? What is the physical purity or genetic purity of any other crops? The same way they ask you the question. See, following group of crops having the same germination percentage. You see the question first. The following group of the crops having the same germination. Options are sorghum, pea, red gram, maize, wheat, maize, rice, bajra, pea, green gram, bajra, ragi, cotton, carrot, cauliflower. If you know it, try to answer it all at least. Anyway, we are going to explain this. So, okay, we are having the one table where we compile different crops and with different germination percentages, right? I am going to, anyway, we are providing all the tables and all the notes, but the extremes you have to remember. The 90%, right? And the 60%. 90% is for maize. Okay, 60% is for the gourds, that what are all the cucurbitaceae family gourds we have, and also the carrot. And 65, right? Then cotton. Here it will come for 85, 80, 75, and 70. Almost the cereals we go with 80 in 85%. We have winter crops, winter cereals, especially wheat and oat. In 80, we have rice, mustard, that is all the brassica species. And in 75, we have sorghum and pulses. In pulses also, you try to remember chickpea comes here. Okay? Chickpea and lentil comes in 80%, but all other pulses will come in green gram, black gram, and also the bajra will come in 75%. Let me know comment section which crops are having a 70% germination. Okay. Oil seeds, not all the oil seeds. So let me know how many of you know which are all the crops are having 70%. So with this, you just try to answer it, which are all the crops having the same. So by saying that, don't get confused. Okay, wheat, maize, rice, bajra, all are cereals. Cereals are, maybe these all cereals are having the same germination. Exactly, it is wrong. So accordingly, if we see that pea, green gram, bajra, ragi, that means millets. Okay, to add there millets having the same germination percentage How much? 75%. Okay, don't forget this. Then they're asking one more question here, the same one. In some of the following crops, the viability of seeds is very poor, and hence the minimum germination is required for certification is 70% only. The crops are. So don't get confused about if they have given certification viability and all. What they are asking is, so it needs minimum 70% germination. So what I left to you is this category, right? What are all the crops will come here? That is the answer. So it is asked in PG, that is JRF exam 2022. So you just answer this. Then, when essential structure of seedlings are missing during the germination test, then we call it as so stubby, stunted, retarded, and damaged. When we call it as stubby, we have a very thick roots or any thick shoots, that is stubby, right? So that it may impair the growth, that is stubby. Then stunted means very dwarf growth. It is not as up to the mark to recognize it as a normal seedling. Then it is stunted. Retarded means the growth, like essential structures are not there. That is why the growth has been retarded. Then damaged means, so the answer is damaged. Okay. The next question is minimum number of seeds required for germination test. This is also a direct question. Try to answer this. Minimum number of seeds required for germination test. Torika answered it yes. It is 400. You have to remember, for some tests they have given working samples, you can't even change that. For physical purity it is minimum 2500, for growth test it is minimum 400 plants. So for germination and for TZ also remember 4 into 100 is 400 seeds. And the next question is standard germination test is conducted under which conditions? Options are optimal temperature and moisture, high temperature and moisture, freezing temperature, and dry conditions. What is the answer for it? Answer this standard germination test. So since we are conducting this germination test in lab and we are going to see the viability, how much, what is the level of viability, we have to provide all the optimal conditions. Then only we can come to know what is the ability of a seed to germinate. So it is optimal temperature and moisture conditions. For some crops, it is light. So the capacity of germination of a seed depends on. It is a very tricky question. Options are seed vigor and seed viability. What is the answer for this? What makes a seed to germinate? Whether it is viability or whether it is vigor. What is the capacity to germinate? Right? Is seed viability. So what is the difference between viability and vigor? Viability means whether it is live or alive, right? If it is live, it will definitely it will germinate. But in vigor, we are going to see, okay, it is live. How much it is live? I mean the degree of liveliness. How much it can sustain in a favorable or wide range of conditions, how much it can sustain. That is vigor. Vigor means its ability or the degree of liveliness. The viability is whether it is live or alive. That's it. Okay. The capacity to germinate will directly depend on the viability. So that third test is moisture testing.
Okay, so moisture testing. So we have completed the germination, get the moisture purity. The third test is like moisture testing. So in this, again, what are important? First thing, the working sample is important. The second thing, the methods of moisture testing is important. The third, the procedure for estimating the moisture content. These three things are important. First, so let's begin with the method. Okay, let's start with the method. Now tell me, for estimating the moisture, which method is commonly used? So if I ask you in this way, like which is like ICAR recommended method for the moisture testing, can anyone answer quickly? Yes. This is the like ICAR recommended method for the moisture testing. it is called as air-worn method. So this air-worn method is again of two different types based on the temperature and the timings. It is a low constant temperature oven method, LCTOM, and high constant temperature oven method. Okay, so this is low constant temperature. That means the temperature here it is like 103 degrees Celsius, and here in this case it is like 130 degrees Celsius, and this is for 2 to 4 hours, and this is for around 17. These are the two different conditions for the LCTOM and HCTOM, high constant temperature method and low constant. So the what is the method? The name of the method is hot air-worn method. So if I ask you like, sir, what is exactly is the air-worn method? Air-worn method is nothing but if you have to check the moisture content. See, what is moisture testing? Moisture testing is checking the water availability or water, or what can the other word? It is like moisture in the seeds, like how much moisture the seed has. That is the moisture, right? Moisture testing, right? That is what we are doing. So in order to do that, what we'll do, we'll take the weight of the seeds before drying it, and then we'll also dry it by using the hot air oven with this temperature. And after, see, once the seeds are dried, the moisture is been removed. So now again we will weight it, and the difference in this weight of the seeds before and after giving it for like hot air oven, that would be the moisture content. The amount of loss of water will be the moisture content that it has. That's how much simple to understand the moisture testing. Okay, and when it comes to the low constant, these are two different conditions that you have to remember. So this is like low constant temperature oven method, and the other one is like high constant temperature oven method. There are other few things like which are very much important is about the process. See, the process of moisture testing is like two different types. One is like one stage process, and there comes the another one, it is called as two stage process. What is the difference between these two? See, one stage process is a simple method where we take the sample and we weigh it before drying, and weigh it weigh it after drying. So based on whatever values you get, so we conclude or we calculate the moisture content. That is what one step or one stage process means. But in the two stage process, there is a one more step which is added, which is like pre-drying. So one step pre-drying is added, and in the second step, the same process which we have followed above will be followed again. So the meaning is in the two stage process, we are going for the initial pre-drying. So you might be already knowing it, like why we have to go for pre-drying. Pre-drying is necessary in such cases where the moisture content of the crop is more than the prescribed temperature. Sorry, prescribed content. The moisture content is more than the prescribed. Where the moisture content is too high, then we have to initially go for the pre-drying, and then only we will be able to actually estimate the moisture content. Okay, so these two steps are involved in case of two-stage process, and in the one-stage process, it's directly like take the sample, weigh the seeds before and weigh the seeds after, and calculate the moisture content. Okay, and here there are few other things which are like first thing. You ask me like, sir, when pre-drying is necessary? See, generally speaking, the cereals moisture content, the ambient or the prescribed, the optimum moisture content like 12%, and for oilseed it is 7 to 9%, and for vegetables it is 8 to 9%. If the moisture contents of the cereal, take any of the cereal, for example rice, if the rice moisture content is more than 13%, then in this case we have to go for pre-drying, and then only we can estimate the moisture content. First step will be pre-drying, and the second step will be the same as taking the weight before and taking the weight after. So now this is under a two-step process. So in this moisture content, apart from this one stage and two stage, there is one more thing which comes into play. One is grinded seeds, and the other one is non-grinded seeds. So what are these grinded and non-grinded seeds? The grinding is just like crushing the seeds before estimating the moisture. So now these are two conditions where for certain crops grinding is compulsory. For certain crops the grinding is not required. So for which crops the grinding is compulsory? Crops Which crops grinding is compulsory? The grinding is compulsory for those crops which are of large size and have a hard seed coat where the moisture loss is like difficult. See, if the seed is such that like the size is very large and also like the seed coat structure, the seed coat structures are such that the escape of moisture is too much difficult, then we have to break the seeds, grind it, and then we have to estimate the moisture. For those such seeds, grinding is compulsory. For the other category where the content is like seeds are very small and the seed coat structures are also not stringent, then in this case the grinding is not compulsory. Okay, this is what you have to remember from the moisture content. Rest will explain. Okay.
You understood it, right? What are the conditions for pre-drying and when we go for a pre-drying? I just want to add one more point here. So in case of one stage or and two stage determination we have, right? Two stage means where we are going for pre-drying first. Pre-drying means we are drying the seeds before estimating the moisture content through hot air oven method. So in this pre-drying, we are drying the seeds at 130 degrees Celsius for 5 to 10 minutes. Only one exception they have mentioned, that is for maize. It is 70 degrees Celsius for 4 hours, 4 to 5 hours. So remember this. Usually pre-drying means just for 5 to 10 minutes we are drying the seeds at 130 degrees Celsius. Then we are just cooling it. Then we are estimating it through hot air oven method. So we have, for reporting the results, we have different formulas for one stage and two stage determination methods. If it is one stage, that means we are directly estimating the moisture content through hot air oven method. We have formula like M2 minus M3 divided by M2 minus M1. So what is M1, what is M2, and what is M3? In hot air oven method, we are using some non-corrosive metals or any bottles, right? Where M1 is the empty container or that empty container with lid, and M2 is the container plus seeds before drying, and M3 is the seeds after drying. So this is the formula. We are going to report results up to 1% decimal. Okay, 1 decimal. In case of two-stage determination where pre-drying is performed, this is the formula. S1 S2 minus S1 into S2 divided by 100. Okay, what is S1? What is S2 here? S1 is the moisture content which is lost during the first stage, that is during the pre-drying. Okay. And S2 is the moisture content which is lost in the second step, that is oven method. Okay. We are taking the mean of it and we are going to calculate through this formula. The same way we are going to report up to 1 decimal point. This is the reporting of results in case of moisture test. Okay. So here if you see the submitted sample for moisture testing, it is 150 grams. 100 gram is for grinded seeds and 50 grams is for other seeds. That means non-grinded seeds. And for working sample, it is completely depend on diameter of the container we are using. If we are using the containers with a diameter of more than 5 centimeter and less than 8 centimeter, then it is 4.5 grams with less or minus 0.5 grams. If it is more than or equal to 8 centimeter diameter, then we are using 10 grams with less or minus 1 grams. Okay. The important one here is 0.2%. What is it? Whatever results we obtain between the two replicates, it should be within the variation of 0.2%. If it is showing more than the 0.2% variations, we have to repeat the process. Okay, and some in tree species it is very difficult to obtain that much, very less variation, right? In large tree species we are getting, so the tolerance is up to 0.2 to 2.5% for tree species. You just make note of it. Usually it is zero point 0.2, but for exception is for tree, that is up to 2.5% can be tolerated. So the working sample should be weighed up to the minimum 3 decimal places. Okay. So the ISTA recommended only hot air oven method, but what are other methods we have? So the reference method like phosphorus pentoxide and Carl Fisher method. This method is considered to be most accurate method, and this phosphorus pentoxide, for calibrating the other methods we are going to use this. Okay, and for routine methods like hot air oven method, this is the most important and the only method which is recommended by ISTA till now. And from some others like calcium carbide reaction method, infrared moisture meter methods, we are having different types of moisture meters also, which is very quick, but it is not much accurate when we compare this with the air oven method. Also we are based on the relative humidity and nuclear magnetic, this NMR method we call it as, and also these some electrical moisture meters also we are having. These are the different other methods we are having for moisture testing. Okay, as sir said, where grinding of seeds are compulsory when we have large seeds and also the seed coat which will impede the water loss from the seed. Then we have to grind those seeds compulsorily. There are the list of crops we are going to provide you where the ISTA recommended grinding is obligatory. Okay, if grinding is not possible for large species, it is not possible. Then we can go for cutting. The size should be more than or equal to 7 mm. Okay, when we follow the cutting.
So here, like I saw one comment. Saurika, you mentioned that soybean is 97 percentage, right? Soybean, sir, it is 97 percentage. Like, what is the reference that you mentioned? Around 97 percentage. Just confirm, clarify. What is the source for that? Where you have seen it is like 97 percentage. That's what I already told you. I told you everyone, like that's not the authentic source. I told you the authentic source. So like what is the authentic source to know about the IMCS standards? Yes, so recall, like which book should you refer to know about the germination percentage, to know about the physical purity percentage, to know about genetic purity percentage, which is the final one to refer, which is the authentic one. Yes, I need from you to have to reply this. Which book is the authentic source to know about all the standards? Not ISTA, please don't get confused. Right, see, ISTA is for seed testing. Is for seed testing. Okay, it gives the process or it gives a procedure for the seed testing. But the standards that it does, certain standards that are given, it is like IMACS. It's like certification standards. If the seeds meet certain standards, certain percentage, only then it has to be certified. That is given in the IMACS. So you have to. So my voice is very low. Okay, is it for everyone? Mansi, like Mansi is saying, like my voice is low. Just confirm once quick. Am I audible to you all, right? जल्दी confirm कर लो. Confirm me once. Am I audible? Yes or no? Okay, fine. Why are you taking so much of time? Okay, fine. So what I was saying, like you have to refer the MACS for germination percentage, for genetic purity, for physical purity, and for also the moisture content. these things have been clearly mentioned. But about the pure seed, the inert matter, and also the other crop seeds, the components of physical purity, and also for the field standards. What are the field standards? Like isolation distance, designated diseases, designated weeds. Everything is clearly given in the IMSCS, which you have to follow. If you can't follow the IMSCS, then just follow the AgriAddict. That's the best thing. The best alternative that you can do. You can follow us. You can just refer the notes that we have provided in our online master course. So same things that we have provided, and they are the most accurate ones, right? So anyhow, like we will be completing the entire seed testing in this video only. No matter like how long it will take, like 4 hours, 5 hours, we will be doing it, and we will be completing it before sleeping. So also like we'll be also providing the timestamps for the different tests. So whoever is interested to see whatever test, you can go out there and see. And if you are definitely wanting to be in the live, you can be on the live and track it. Okay, so this is about the things like that you asked for. Soybean is not 97. That's why I told you in the starting, in the seed technology, the problem with this is the content. The content, it is mentioned like so many different kinds of answers, like errors have been mentioned in different books. When you compare the different books, the same content has been given differently in different books. That's the problem. So that's the reason I told you initially, like ISTA and like IMSCS is Bible for the seed testing, and for certification, the IMSCS plus the seed act and the seed rules and seed control order, and these are like Bible. Okay, so that's what like most of the things will come from. Coming back here, one thing I have to mention. So I told you about the grinding part and non-grinding part, right? Grinding is compulsory for the seeds which are large, and for the seeds which where the moisture loss is not easy, right? So again, in this grinding, we have two different types. One is coarse grinding and one is fine grinding. Generally speaking, I've separated it to remember in a better way. The coarse grinding, it is for legumes, okay, and the fine grinding, it is for cotton and the cereals. Just remember these three different areas. The rest you can forget it. No issue on that. See, when it comes to the legumes, the condition is coarse. When it has undergone the coarse grinding, the 50% should pass through the sieve. Sieve means what? The pores. Pores of 0.5 mm and not more than 10% should remain on a 1 mm. When the pore size is increased, should not, like more than 10% should not remain on it. Everything should pass. But when it is a sieve size is 0.5, then at least 50% should pass. Okay, this is the criteria for the coarse grinding. In case of fine grinding, how much fine grinding it has to be done? It has to be done in such a way that 50% should pass through the 4 mm. Okay, through the 4 mm, 4 mm, 50% should be passed. Okay, so this is about the fine grinding and the coarse grinding that you have to remember from the moisture estimation. So where we have completed most of the things, we have clarified and cleared most of the things from the moisture testing too, and this is where exactly the questions will come from. I bet in the exams there is nothing to be asked apart from what we are discussing. Most of the things will be very clear in terms of seed testing part from these things. Fine. Okay, let's move. This question discussed. Okay. So for determining the seed moisture content in the pigeon pea seeds, the seed sample is dried to O1 at a temperature for the duration of. For the pigeon pea, what you have to understand is whether it is HC high constant temperature O1 method, or it is like low constant temperature O1 method. Can anyone tell me what is it? Whether it is high constant or low constant? Yes, anyone? So it is already been mentioned, like it is like high constant, but the hour varies. As I told you, it is 2 to 4 hours, but still the conditions are different for different crops. Fine. So you have to try to remember at least for the major crops like cereals, major pulses, major oil seeds. That will definitely help in the exam. Okay. Next thing, the maximum moisture content for the safe storage in the wheat. What is the maximum moisture content for the safe storage in the wheat? So what is it? Yes, anyone? I mentioned about the safe storage. I mentioned it. So for cereals, generally speaking, remember the general things. Generally speaking, it is like 10 to 12% for cereals, and if it is for oil seeds, it is 8 to 9%, and if it is vegetables, is 7 to 9%. And in this case, here the maximum moisture content that is safe for the storage of wheat is 12%. Now you can ask me, sir, then 10 might be also the correct answer here. Why the 10 is not the correct answer, 12 is the correct answer? Because they have asked about the maximum. The range is like 10 to 12, right? In the upper limit, it is like 12. 12 is the safe storage for safe moisture content for the storage in case of wheat. You have to think before answering. Don't get confused by yourself. Moisture content, seed moisture content varies from crop to crop, and which ranges from. The range is commonly from 9 to 12. What we have seen, and it can be like 7 to 12. Don't get confused. This is just a object type question. Just try to answer. That's it. Don't get deep into this thing. Generally speaking, the first one is right. The next, ISTA recommended method of seed moisture determination. So there are 3 methods which have been mentioned here. Just tell me like which is the method which is recommended by ISTA. Quick. Yes, it is obviously the AR1 method. See, even if they give like method, if they give drying, anything, if the word is with the AR1, just go for it. That's it. Don't get confused about anything else. Fine. And again, you have to remember, if you have to recall all the things, it is like HCTOM and LCTOM, that you have to know that this temperature is 130 and this is like 1 knots. Okay. Fine. 130 for like HC2M and okay, see this is 103 for like LCTOM. Fine. And about the duration, it is for 17 hours and it is for like 2 to 4 hours. That depends and that varies from the crop to crop. And again, other things what you have to remember about the grinding, which I told you. And again, in the grinding, you have like coarse grinding and you have like fine grinding. Okay. So next, you have like in the analysis, you have like 1 stage process and you have 2 stage process. So what is the difference between 1 stage and 2 stage? 2 stage is like pre-drying is added in addition. So when the pre-drying is compulsory, again, I told you already, when the moisture content is high, then the pre-drying is necessary. That's it. So entire moisture content things in a single slide. Okay. Safe moisture content for the storage of seeds in cereals. Same thing. So it is 10 to 12%. During moisture testing, choose the correct option for the following statement. So 100 grams for the grinded seeds, 50 grams for the other seeds. So these things are also clear. We already told you it is 100 for the grinded seeds. The what they are okay. So this is about the samples which have been taken, submitted sample. So this is question which is related to submitted sample. If the sample is grinded, then we have to submit 100 grams. If it is not grinded, then we have to submit like 50 grams. Okay. The next, the fourth one, it is like viability test. We have completed first one, we completed the physical purity. Second thing, we completed the germination. Third thing is we completed the moisture testing. The next test is viability. So what is viability test? The viability test tells us whether the seeds are alive or dead. Based on what? That depends on the method, right? That depends on the method, which principle, based on which principle it is telling us. But what exactly it will tell us? It will tell us whether it is alive or dead. That's it. That is about the viability. So whenever we talk about viability, we only talk about the one prime test, which is like TT's test, Z test, like topographical heterosolium test. So next, the other than that, do you know any other test which is related to the seed viability? Have you come across like embryo excision test? This is also one recommended method for the seed viability testing. So we also have one more method. What is it? So it is the second method, embryo excision test. And third, we also have one more, which is like X-ray test for calculating the seed viability. From the viability test, what we will be knowing? We will be knowing about whether the seed is alive or not. That's it. Okay. what is the importance of this TTZ test? So this TTZ test, it is being accredited by ISTA, the standard test by the ISTA for both viability and vigor. Now, again, there would be one more confusion. Sir, this TTZ test, it is only for viability or it is also for vigor? So this TTZ test, it is not only for the viability, it is also for the vigor and the viability. So now you can ask me, sir, how come like the TTZ tells, just a minute, how come the TTZ test tells us about the viability or vigor? So when the Gayatri will explain you the process of the seed viability, like the procedure of TTZ, she'll be able to tell you like how it is also related to the vigor. Fine. So next it is average existence test, and third one is X-ray test. So we'll go in detail. So starting with TTZ test. So it was given by Lacon. This is very much important to remember. It was in 1942. Know the full form and also know about the chemical. Okay. Explain the procedure. Gayatri will explain the process for you.
So when we talk about viability test, whatever we got into mind is TTZ test, that is topographical tetrazolium test. Why it is called topographical means topography means the location. Tetrazolium is a chemical, right? What is the chemical we are using it here? 2,3,5-triphenyl tetrazolium chloride. Okay, that is why it is tetrazolium. And why topographical means we are evaluating the stains, that is based on the location. If it is completely staining the embryo or endosperm or radical, we are seeing those essential structures, whether they are completely stained or incompletely stained, or if they are not at all stained. So since we are assessing the viability based on the staining pattern and location of the staining, that is why it is called topographical, and the chemical is tetrazolium chloride, that is why it is called as TTZ test. So what is the working sample for it? As already mentioned, is 400 seeds, that is 100 into 4 replication. It is recommended by ISTA. Remember this. What we are doing here, so we are just imbibing the seeds. Okay. Before that, we are just moisturizing the seeds. Okay. We are imbibing the seeds for one night usually, that is 7 to 10 hours. Then we are dipping the seeds in this salt. Then what happens? What is the principle of TTZ test? This chemical, the 2,3,5-triphenyl tetrazolium chloride, is non-colored. Okay. It is a colorless solution. When the seeds will accept, or when they have dipped into this solution, since seeds means is a live entity, right? What happens inside? The enzyme, it is dehydrogenase. Okay. The name itself, it indicates dehydrogenase means which the reduction of hydrogen takes place. So this enzyme will react with this chemical, that is tetrazolium chloride, which is a colorless. So it will replace this chloride ions with the hydrogen ions. Okay, then it will form a one stable component, that is formazine. Okay, and it will give a red color. Means when this colorless solution will turn into the red colored one, then we can say that there is an activity of dehydrogenase enzyme. The activity of dehydrogenase enzyme is high in live seeds. That is, the seed is alive. That means they are viable. Then only this formation. That means that is based on the staining, right? If it has taken red color staining, that means there is an activity of dehydrogenase enzyme, and based on this we can categorize it as a viable or non-viable. And as sir told how it is used for vigor, that is, you can see here, I'll tell you, all these starting 4 diagrams will depict the viability. Okay, you can say that you may ask that since it is unstained here, so the important part like radicals and the radical plumule axis, everything whatever we to consider it as a viable, the essential structures like this axis, radical and embryo, has got stain. That is why they are viable. Here, even the majority part of the stain, the very important is tip of the radical, your whole radical, your radical with plumule and axis, even the embryo part, and since it is unstained here or incompletely stained, those essential structures, they are called as non-viable. Even these also, you can see here. The very essential part of a seed is radical first, right? Then that axis, right? Then embryo. Everything, it is completely unstained. So these are all non-viable seeds. Just forget about non-viable. Think about viable. You can see here it is completely 100% stained. And it is a little bit. And here, so based on this, we are considering all these 4 seeds as a viable only. But based on the intensity, how much intense and what is the, based on this staining pattern, we can categorize this into, it is a high vigor seed, right? Whichever is showing a complete 100% intensity, that will show high vigor. It follows like this. So this test based on the intensity of a color, we can use it for vigor test also, and also for the viability. Got it? And other than that, we have embryo excision method and X-ray radiography. When we go for this method, in tree species, we have few species which will take almost 60 days also to get germinate. In those cases, we can't wait for the result till 60 days, right? So in that situation, we will excise the embryo. Okay, the TTZ, let me add one more point here. The TTZ is more preferred, recommended, and it is called quick viability test. Why so? In other methods, as I mentioned for embryo excision and all, it is where the tree species will almost take a 60 days. But since it takes only 4 to 6 hours based on the crop species, that is why it is called quick viability test. 4 to 6 hours is very feasible, right? That is why it is quick and it is recommended and more widely used. In embryo excision methods, we are going to excise the embryo. How? Same way, we are moistening the seeds or imbibing the seeds first in running water. Remember this. So that whatever the hard seed coat will be broken when we keep the seeds in a running water at almost 20 to 25 degrees Celsius temperature. Then with a sterile environment, like we are using 70% ethanol, we are going to excise the embryo from a seed. Then we are keeping it for germination. The maximum of 14 days. So based on, at the end of the day, based on the pattern, if they are viable or non-viable, based on the germination capacity, we are going to distinguish. Okay? This is embryo excision method. What happens in X-ray radiography? The same way, after the imbibition of a seeds or pre-moistening the seeds, we are going to dip these in a barium chloride solution. Okay. And then the seeds will be exposed to X-ray. What is the principle behind this? This barium chloride solution will penetrate into dead cells. Since it is a semi-permeable, it will penetrate only into the dead cells, not into the live tissues. So that when we expose these seeds to X-ray, in where we will get a patterns, right? Wherever we have got a pattern, that is dead, since barium chloride will penetrate only into the dead cells. Okay, based on this, it is also used for tree species, where tetrazorium chloride test is not feasible. So majorly for the tree species, we are using these two methods, embryo excision method and X-ray radiography methods. So the questions are, TZ test color of living tissues of seed will change to. As we discussed already, so this TZ chemical is a colorless one initially, but when it reacts with the living tissue, it will form into red colored stable compound, right? That is formazine. It is red color. The next question is the quickest seed viability test. Options are T test, Z test, germination test, and TTZ, TZ test or TTZ test. What is the answer for it? As we already discussed, since it requires only 4 to 6 hours for evaluating, the quickest is TZ test. The next question is first step in seed viability test. Imbibition, staining, and evaluation. Try to answer this question. It is very important. First step in seed viability. We'll directly go into staining, right? Because whenever we remember TZ test, we will remember the staining pattern, and based on the staining pattern, how we were going to evaluate. But the thing is imbibition. So first in TZ test, we are going to imbibe the seed first. It's pre-moistening the seeds. After TZ, let me add one more point and make note of it. After dipping into the solution, to make embryos very clear, right? We are dipping these in a lactophenol solution. Remember this. What is lactophenol solution? That is lactic acid and phenol. Obviously the water and glycerol. Okay. That is in 1:2. Is lactic acid, phenol, water and glycerin. Just remember this. To make embryos clearly visible, okay, we are using this solution in TTZ. Don't just confuse with, okay, where we are getting this lactophenol solution, because there are so many questions asked about this one. Right? People are answering it. So the first step is imbibition. And this other question, which of the following seed testing method is used to assess the seed viability? Tetrazolium test, KOH test, and top of the paper method. It is tetrazolium test. We are going to discuss this KOH test also. Okay, the tetrazolium test will determine the level of activity of which enzyme? The options are ATPase, dehydrogenase, carboxylase, and isomerase. Answer this. Tetrazolium test. Okay, right? It is dehydrogenase. So, in short, just remember TZ means which chemical we are using, what is the color of it, what is the principle, that is which enzyme is responsible for changing the color, and the duration and the steps. And don't forget this lactophenol solution. Okay, just that's it. It's about the viability. Since we have only one recommended test for seed viability, that is TTZ test, remember that. The other question is seed viability test used in tree species is X-ray photography, TZ, and embryo excision methods. Answer this. X-ray, TZ, and embryo in tree species, which are the methods we are using? Okay, this X-ray and embryo excision only. We are not going to use TZ in tree species. Okay. Okay. The which chemical used for biochemical test for the seed viability? Which chemical? That means the tetrazolium test. Which chemical we are using in TTZ test? That is 1% tetrazolium chloride. Okay, just remember here about the concentration. There are two conditions. If we are bisecting, if we are not bisecting it, okay. If we are not bisecting the seeds, means we are not cutting or we are not piercing it or anything, just we are directly using the whole seed, then 1% we are using, okay, 1% tetrazolium solution. If it is bisecting, 0.1%. Remember this. Just like this directly you can just go for 1%. If they specifically mention for bisecting, then you have to choose 0.1%. Okay.
So we completed the viability test. So a few things to add, like one, I'll add a few important things. Okay, to remember it easily. Things like we talked about aliveness. Gayatri told about the TT that test, which is like most important for you to remember. So in this, we talked about one enzyme which is dehydrogenase. So what exactly, like how we are going to relate this dehydrogenase enzyme to the aliveness of a seed, whether seed is alive or dead. So how we are going to correlate it? To understand in simple words, so how can I say that Gayatri is alive? How can I say it? It respires, right? It's all about respiration. Similarly, in case of seeds, how do we say whether the seed is like alive or dead? It's based on the respiration. When the seeds are respiring, enzymes will be definitely active. So in that, there is a one such enzyme which reacts with this TTZ, that is tetrazolium chloride. This enzyme reacts with that. That's how we are able to know indirectly that yes, the seeds are respiring. Based on that, we come into the conclusion what is the viability percentage of seeds. That's it. Viability of the seeds, whether the seeds are viable or not. That's okay. This is about the TTZ that you have to remember. The principle is important, and also like whether for the concentration for the bisected seeds and the non-bisected seeds. Both the things are important for you. Like 1% and the 0.5%. The next thing. So the fifth vigor test that we have to discuss. So it's about the vigor test now. So earlier it was like viability, now it is for the vigor. So now before directly jumping into the vigor, I'll tell you the importance of this. Why exactly? What are these vigor? For example, it has been observed. It was been observed before like establishing the vigor test. It was been observed that the seeds which had like germination percentage. Let me take the two seed lots. The seed lot 1 and the seed lot 2. Consider two examples. So the seed lot 1 germination percentage when it was calculated, it was 98%. And the germination percentage, I'll just mark it as G1. G2, which was tested, the germination percentage, it was also like 98. That means like both seed lot 1 and 2 had a same germination percentage from the lab results that was taken in the lab. Okay. Now, when this particular seed lots, the seeds from the seed lots are given in the field conditions. They have been sown. Okay. They have been sown in the field conditions. There, the germination one was like 90. It was not matching with the above one lab conditions. The other one was around 60. So why there is a difference here? What is the reason? Because if in the lab the germination percentage is 98, both are 98, then exactly both should behave similarly. If it is 90, the other should be also 90. And also like how come it is 90? It should be 98, which is 1%, 2% plus or minus is fine. But how come is 90% and how come in the second case it is 60%? What is the reason for this? Simple reason for this is the germination test. When we are going to do the germination test, it is in the lab. That means it is in the favorable conditions. We are doing the test in the favorable condition, right? But field conditions are not always favorable. Is like very dynamic. So now this germination percentage, which was earlier known to like evaluate the potential of the seed lot, is not at all useful right now. Yes, it can say about the germination percentage, but only in the favorable conditions. In the field, if the favorable conditions are there, then the percentage, the accuracy will be same or it will be more accurate. But what if the conditions are adverse? For this, there was a concept of vigor that came into play, which will evaluate the field crop stand or the field potential of the seeds based on whether they can survive on the extreme condition or not. Okay, and this term, the vigor term, was given by the Frank. How vigor testing came into play. So this thing, being a student of seed technology, you have to know. So coming back, these are the tests. See, the test of the seed vigor test can be classified into performance based, it can be divided into stress based, and it can be divided as like biochemical based. See, these are the list of tests. You just remember the list of tests here that are given here. I'll give you a brick gravel test. Who gave this? It was given by Iltner, right? It is also called as Iltner test. Cool test, it is commonly for cotton. How to remember? And this is for cold test, it is for maize. Again, simple trick. Cool, O, cotton, O. Remember, cool for cotton. That's it. And next we have accelerated aging test, controlled deterioration test, and we have again TZ test. And this is for ISTA recommends this test to evaluate the vigor in case of soybean. And we have like RQ, respiration quotient test, and then membrane integrity test, and the GADA test. This is in the biochemical test. These are the different categories. In that, like whatever is like mostly important, I have told you about. Next, you just try to go it in bit detail and try to understand it. And one more most important thing from the vigor test that you have to remember is about the ISTA recommended again. See, in every test that we talked from the starting, the physical purity, the germination, the moisture testing. So in all the things, like what is important, it is much more important, and it's mandatory for you people to know which tests are ISTA recommended from different one. Okay, so here the ISTA recommends the conductivity test. It is also called as electrical conductivity test. Do not get confused. It is one and the same. Next, we have accelerated aging test. We have controlled iteration test, radical emergence test, and we have tetrazolium test. So this is 1, 2, 3, 4, 5. You have to remember this 5 tests which are the ISTA recommended vigor test. And also equally it is important to know about the crop. Crop is also important. See, accelerated aging test, it is easy. It is for wheat. And controlled iteration, it is for brassica. Radical emergence, earlier it was for maize, brassica, and Raphanus sativa. Now recently the wheat has been also included. And the last one, the tetrazolium test for the vigor estimation, it is for glycine max, which is soy. That you have to remember. So index 1 and 2. So now Gayithri will explain you about the what is seed index vigor. So seed vigor index 1 and what is seed vigor index 2. This is also another important thing that you can directly get in the exam. Okay, so let me do it. The seed vigor index 1. This is just an index that is being calculated using one of the vigor parameters. Seedling vigor index 1. And I'll also keep it at 2. What is the difference? It is just the germination percentage into length divided by 100. Okay, and the second one, it is the germination percentage into seedling dry weight divided by 100. That's it. See, the difference between the vigor index 1 and vigor index 2 is with respect to the seedling. Here length and it is dry weight. Seed 1 is with the length in terms of centimeter. Remember the unit also centimeter, and it is in the gram. Seedling dry weight mg, milligram. This is in the mg, milligram. Okay, so this is about the seed vigor index 1 and 2 that you have to remember for your exams. Rest, most of the things are very clear. And the other tests, there are several tests that are available for the seed vigor, not only the one which I have mentioned, but you have to go through all the tests. But the detailed procedure of the individual tests are not required. But still, if you are able to at least remember 1 to 2 to 3 lines about each and every test, at least the basics about it, that will help you in the exam. Okay. The next question. So let's see the questions. The term seed vigor refers to what? The ability of the seed germinate under optimal conditions, and environmental factors, so genetic purity. So here, what does the seed vigor, the term refers to? The options are not mostly relevant. So the term seed vigor refers to the capability or the potential of the seeds to germinate in the stressful condition or adverse condition. Why it is like stress or adverse conditions? When the conditions are favorable, all seeds will germinate. All seeds will germinate which are alive. Only when the conditions are unfavorable, only those seeds will germinate which are having like high vigor. That's why vigor definition always says about the adverse conditions or the stressful conditions, about the seed vigor. And vigor, and also don't confuse with the term. It can be written as GOR, or it is also written as GOUR. The cold test, as I told you, the cold test is used to assess what? Moisture content, no. Assess the vigor, yes. No. Dormancy, seed viability, no. So I already mentioned, it is like the cold test. It is in the maize to evaluate the seed vigor. Okay. So next we came to the next one, which is like genetic purity. Another very important test in case of seed testing. But I'll also clarify one more thing. We came across like 7 different tests. So now tell me, like the 7 different test procedures that we have during the seed certification, which is being done, do we actually go for all the 7 tests, or do we only go for few of them? What exactly? How it is? Anyone? Yes, Saurika, Mika, Mansi, anyone? Do we actually go for all the 7 different categories of seed test, or we only do some tests? Yes, exactly. We only go for routine tests. So for me, can you tell me like what are the routine tests that we have? Is it correct? These are the routine tests that we have. So here one more is also included, which is like other crop seed test for other crop seeds. Germination percentage, mostly like most commonly these 3 are being tested, plus one more you can include, which is test for knowing the other crop seeds. Okay, so these are the routine tests that are being followed in the seed certification out of 3 tests. Now then, what about the genetic purity, where it is important, where the genetic purity test is conducted, where exactly? So you have to remember, when we talk about genetic purity, so here this is also called as testing for genuineness of variety, or the test is also called as varietal purity test, VPT, or it is called as genetic purity test, or simply it's genetic purity test, genetic purity. So all these are for the same thing, same test, where we are trying to know its genetic content, where they are genetically the seeds in the seed lot are genetically same or not. Fine. So for this, we only do it for hybrids. We do not do the genetic purity test for the varieties. First thing, the first condition, it is only done in the hybrids. So next thing, whether it is done in all the hybrids or only few hybrids. So again, the second thing, it is only done in the few hybrids, not all the hybrids. Okay. So 2 things are clear. And the third thing, which test is used to calculate the genetic purity? So in India, we only use GOT. See, you might have heard like as a synonym. Wherever you see the genetic purity, you see the GOT. Wherever you see the GOT, you see the genetic purity. It is used as like synonym to one another. It's because of the fact that the GOT is only GOT is used in India to calculate the genetic purity. But the only process, it is the only process that is available? No, there are also the other processes which are available to evaluate the genetic purity. So how the genetic purity is generally evaluated? So it is evaluated by in majorly 3 different ways. If I have to evaluate the genetic purity, how I am going to do it? A simple process to evaluate the genetic purity, or to know whether, if I have 2 seeds, if I have to know like whether the 2 seeds are similar to each other or not, the simplest and most accurate thing is I have to sow both the seeds, and I have to see, starting from the sowing till the emergence of the plant until the end of its reproductive stage, I have to see for all the characters, for all the duration, for different stages. I have to see with compare one and the two, and see like if there is any difference in any of the steps. This is the most accurate method to know about whether 2 seeds are genetically pure or not. So how it can be done? It can be done with the lab experiments, or it can be done in the greenhouse, or it can be directly on the field. So in the field, how it is generally being done? It is simple. We have a seed lot. Seed lot will be of like any kg, any number. So in that, we'll take the working sample. What is a working sample for genetic purity? That is like 400 seeds again. 400 seeds, like 100 seeds in 4 applications. So we have to take it and we have to go for sowing. So where we are sowing, as I told you, like we use only GOT. GOT is like field test, where in the field we will be sowing the seeds and we'll be checking for all the stages if there is any difference, and for all the morphological changes and visible changes that we can see, we'll observe it, we'll compare it, and that's how we will get this one, the genetic purity percentage. And I told you about the working sample. But in the genetic purity, along with the working sample, the other important things to be known is about the submitted sample. I'll talk about that. So other than before that, in the lab examination also, like there are different categories. Like one is like examination of seeds directly, just like the purity, physical purity examination. We can directly examine the morphological differences between the seeds if there are any. Based on that, we can separate the seeds. Next thing is examination of seedlings. In case of seedlings, we have like a specific coleoptal color in case of cereals. Based on that, we can differentiate the varieties. And based on the apocotyl color in the beta species, based on the cotyledon color in case of brassica, and based on the fluorescence color, we can differentiate between the festuca and the lolium, right? So these are certain specific methods that are available for certain specific crops. Next, we have the chemical test. In chemical test, we have phenol test, modified phenol test, seed peroxidase test, KOH bleach test, that is for sorghum, and peroxidase is for soybean, parasulfate test for the paddy, and NaOH or potassium dichromate test for the pea, wheat, and alcohol test for the lupins. So these are some of the again specific tests that are available for different crops. Do not get confused with that. See, you might get a lot of tests, the name of the test in the while reading the CTEC. So what first thing you have to remember, what first thing you have to do, you have to see that this test is for what purpose, whether it is to calculate the viability, whether it is for vigor, whether it is for germination, whether it is for genetic purity, or whether it is for physical purity. Only then you will be able to recall and decode the things. So now wherever you see, if you see the phenol test, you should be remembering that it is for knowing the genetic purity. If you see the coleoptal color test, you have to quickly remember that this is under like genetic purity testing. Okay, so there are the methods which are available to test the genetic purity, but for the seed certification in India, we do not do any of these things. We only do GOT, and again, it is only for the hybrids of few crops, not for all the crops. Okay, and we have like biochemical tests, which is electrophoresis, UTLIF, page, SDS page. So you can remember, you have to remember actually, it's not you can, you have to remember this if you are giving your ASR minute exam or the ICAR PhD exams. All things are very much important for you. So now the question is like which of the following method is used to check the varietal purity? First thing, germination test, growth test, viability test, vigor test. So obviously like, you can mark it as growth test. Okay. Next, the seed coat peroxidase test is used in which crop? So it is used in soybean. Next, the GOT is compulsory for the certification of the following crops in India. See, this is where I told you that GOT, the genetic purity testing, it is compulsory for only few crops in India that are also like hybrids, not for the varieties. Only for the certain hybrids, this GOT is compulsory. For which of the crops GOT is compulsory? The GOT is compulsory for cotton, it is compulsory for castor, it is compulsory for tomato, it is compulsory for brinjal, and it is compulsory for muskmelon, and it is compulsory for TPS, 2 potato seeds. So it is compulsory for cotton, castor, tomato, brinjal, and muskmelon and TPS. Why it is so? Why it is only for these crops? What is the common thing between these crops? Common thing is respect to the method of hybrid seed production, how they are being prepared, how the hybrids are being developed. See, if you know about the cotton, if you know about the tomato, brinjal, you know it, right? So this have been with the emasculation and ant pollination. So whenever the things are with the emasculation and ant pollination, there will be always certain amount of error. So if there are certain amount of error which is high, then obviously the genetic purity mixture, the genetic purity will be deteriorated to some extent. That's why we compulsory do this test here in these crops. But what about the other? Like this is also same. And what about the castor? Castor, it is because of the reason why we are going to do it. It is because of the reason that it is with the pistillate line. The hybrid seed production method, it is with the pistillate line. So because of this reason, which are like temperature sensitive, so there is a sex reversion usually that takes place in this castor. So that is why even in the castor we go for the hybrid, sorry, GOT, which is growth test. Okay. So here in the question, then the answer would be which one? From the 3 options. Here the cotton is right, brinjal is right, castor is right, but the wheat is wrong. And for oil seed, no, not for all the oil seeds. Except castor, there are no oil seeds here. The cotton, maize, and bajra, this is also fall. I just given the random options here. So all these are false, like only for these 6 crops, we go for the GOT in India. And this is with respect to the India, remember. Okay. And sample size for the growth test for the jute is. So this sample size is again one of the important thing to remember. So in questions, in the exams, you might get this question. So how to answer this question? So you have to follow this table. So where you will find this table again? Where you will find this table? So you will again find this table in the IMSCS. It has been clearly mentioned. So here 1000 gram, the samples for the growth test to be drawn simultaneously with the other quality test will be sampling. I am talking about the submitted sample, the size of the submitted sample. Size of a submitted sample for the growth test, it is 1000 gram for the few crops, 500 grams for the few crops, 250 grams for the few crops, and 100 grams for the few crops, and the 250 tubers per planting for the sweet potato, and the other vegetative propagated crops. So how to remember this? I have also mentioned the shortcut. Remember this shortcut, CGMS. CGMS, C for cotton, G for groundnut, M for maize and soybean. And not only for these 4 crops, but also for the species of other genera which are with the same seed size. So whatever size the maize has, whatever size the cotton has, whatever size the groundnut and soybean has, with a similar size, if there are other crops, all of them should be the 1000 grams. Next, for the next shortcut is WPS. You might have already used the WPS office for the PDF. You can remember that it is like wheat, sorghum and paddy, and the species of the similar size you can go for 500 grams. That's how you can remember. And 250 it is for the beta species, and with the this is very easy to remember since only the beta is covered. Next, for the 100 grams it is for the bajra and jute and species of the other genera with a similar size. Okay, here it has to be mentioned is for the similar size which is missing. Don't get confused. The last one, 250 tubers per planting. It is for seed potato, sweet potato, and other vegetative propagated crops. This is like another most important thing, which is like size of submitted sample that you can't really skip. Next, let's see one more question. So another very interesting question here. So this is with respect to the for the crop having maximum permissible limit for the off types 0.2%, the minimum number of plants required for conducting the GOT. So they are saying that for a crop having the maximum permissible limit of 0.2%. So what they are saying is, if there are 1000 crops, 1000 plants which are standing in the field, in that 1000 crops, the maximum number of off types, should be only two out of 1000. Okay, if there is more than 2 out of 1000, then it will be rejected. So this is the maximum permissible limit according to the MCQs. That is what they are saying. So if this is the prescribed standard, that is off type should be only 0.2%, that is 2 in 1000. What is the minimum number of plants to be required for conducting the GOT? So what is the minimum number? Like if from the 1000 plants there should be only 2 off types, that is 0.2%. Then what should be the minimum number of plants required? So to test it, how many plants should be planted? So this is with a simple formula called as 4n. See, simply what students do, students usually try to remember this table, which is already given directly in the MSCs. But before this, I'll also tell you the conceptual way of answering, even without remembering the table. So the actual formula is 4n. So what is 4n? So ISTA or IMSCS says that if I have the maximum permissible limit is 2%, 2, that is like can be only 2 in 1000. If there are 2 off types in 1000, it is permitted. If there are 3 off types in 1000, it is not permitted. This is what it is saying. So how many seeds should I plant so that to test the number of off types? So here it says that it should be 4 times this number, total number. So if it is like 2 in 1000, is like 1 in 500. That means like at least 500 plants or 500 seeds should be planted in order to detect one off type. So in order to make it very accurate, it should be like at least 4 times of this 500. Okay, it should be at least 4 times of this 500, where the n value is 500. what is the actual meaning of N now? N refers to number of seeds which are to be planted to at least get one off time. So this is what it means. Like, let me repeat, what is N here? N is defined as number of seeds which are to be planted to at least detect one off time. So in this case, since they have given it as like 0.2%, here, as I told you, in order to detect one off type, minimum 500s are required. But according to the formulas, like the standard is at least it's a 4 times. To make it very accurate, we have to take it at least 4 times. So it is simply 4 into 500. The answer will be 2000. You can apply it for anything. If they give that permissible limit is 0.1%. The minimum number of plants required for connecting the GOT. Then here it is like 1 in 1000, right? So then totally there should be like 4000 plants which are needed to be evaluated, because n value is 4000 here. So 4 into 1000, it comes to the 4000. Same thing if you just try to check this, same thing has been mentioned. I'll just give you the example. See here the permissible limit is 0.3, that means 3 in 1000, right? 3 in 1000. If it is 3 in 1000, just divide 1000 by 3, you'll get around 999, I guess. It is like 333 you get, right? It is like 1 in 333. So if you just multiply it with 4, this is how much you are going to get it. That's it. Okay, so this is how you can remember this even without remembering this complete table. Otherwise, if you are feeling difficult to understand like what I said, so you can just remember the table. But if you are able to understand it, it's better for you, because in the long run you will never forget it. If you just remember the concept, that's for lifetime. Next, the coleoptile color test is for. So I told you already, the coleoptile color is for the cereals. Okay. Minimum seed standards for genetic purity in the castor for the foundation seed production will be. Okay. So here in the genetic purity, most important thing that is usually asked in the exam, it is about the field standards, or it is about the seed standard, the genetic purity standard for the foundation seed and the certified seed. Foundation seeds and the certified seed. So now for the foundation seeds, for all the varieties and the hybrids, whether it is variety or whether it is hybrid, for all of these seeds or for all the crops, the percentage is minimum, it is 99%. The standard genetic purity that has to be maintained or followed when it is in the foundation seed stage for all the varieties and hybrids is 99. But there is one exception here. The exception is like castor, but for castor up to 95% is permitted. So the castor crops has to be maintained with at least 95% genetic purity for this foundation stage. But in the certified stage, it becomes a bit complex here, because for the varieties multi-lines, there is one standard, where for the synthetics composites, there is one standard, and for the hybrids, there is another standard. Okay, so because of this reason, it becomes complex. How to remember it? So you can remember it by this seeing this table. And again, where is this table? This is from again IMSCS. Don't get confused. No need to search anywhere. Just go for the IMSCS. In the foundation, I told you for the foundation it is like 99% for all the crops, but exception is like castor, which is like 95%. And for the certified seeds, in the certified, the varieties, composite, synthetics, and multi-lines. So this comes with the 98%. For all this, it is 98%. And for the hybrids, for all the hybrids, it is 95%. Certified seed production. But for the hybrids of cotton, true potato seed, muskmelon, brinjal, and tomato, which I told you earlier, where the GOT is compulsory, for these crops, it is like 90%. Again, there is one more crop which is left, which is hybrid castor, which is having the least standard genetic purity, which is like 85%. These things are important. You have to remember this table as it is. Okay. So this completes the genetic purity. So next, we will talk about the last one, the other one, which is seed health testing. Just a minute. Okay. Few more questions are left. Like growth test is done for the verification of what? Growth test. Growth test is what? GOT. Just grow it out. That's it. Grow it and know the genetic purity. It is for the genetic purity. Next, the test weight is. What is this test weight? See, the test weight is nothing but the 1000 seed weight. Remember this. 1000 seed. What is 1000 seed weight? Name itself indicates is the weight of how many seeds? 1000 seeds. Remember this. Like whenever you see the test weight, just correlate with the 1000 seed weight, where it's the weight of 1000 seeds. There is one more term which is related to this. It's like seed index. What is seed index? Here the seed index means like it is a weight of 100 seeds. Remember, just like we had like seedling or seedling vigor index 1, 2. We had index here. The formula, if you have to just recall, it is like length, seedling length, and the vigor index 2. It was like dry weight, right? It was in milligrams. Similarly, like whenever you see the seed index, it is like weight of 100 seeds. This is how you have to connect it. Whenever you see some terms, you have to recall like whatever you have understood and whatever you have read from the specific term. Next thing, there is one more match the following. You can match it. So here it is asking for growth test, osmotic stress test, viability test, brick level. As soon as I see the growth test, I can just drop it to test for the genetic purity. As soon as I see the viability, it is for making the test of the aliveness of the seed, and brick gravel test, it is test for vigor, and the last one is left upon tested, it is for the PG and mannitol. Simple. You can match the two, rest you can, they will be automatically matched by themselves. Okay. The last test that is left is the seed health test. So now we have successfully completed, started from the physical purity, we have covered the germination, we have covered the moisture content, and next we have covered the viability, vigor, and genetic purity. The last test which you should be knowing is about the seed health testing. So Gayithri will explain about the seed health.
Yeah, this is the last test, but not least. Why? Because seed health is more important. Okay, let us assume we are having a seed with 100% germination, 100% purity and all, but if it is infected with any seed-borne pathogen, or if it is completely infected with any diseases, is there any use of that seed? No, right? That's why. Same for us, like also being healthy and being fit is also very important. The same thing for seed. Along with the germination capacity, along with having a vigorous capacity to germinate in a field or any stressful conditions, being healthy is also important. So healthy seed is more important. The same concept is, if we see into the history of seed health testing, the first seed health test was developed by Hiltner. It is already mentioned in a vigor test, that is Hiltner given brick gravel test, right? He used it for seed health. How? In seed health, there are so many aspects which will contribute to the seed health. Not only it is free from diseases. If it is healthy, obviously it is vigorous. Obviously it will germinate very well, right? So in brick gravel test, what we are doing, the seeds are covering with the brick gravels of 3 mm thickness. Okay. So based on that, if seeds are penetrating these brick gravels, we are consider them as a vigorous. When they can penetrate, if they have enough potential, then only they can penetrate, right? So what he observed, the Hiltner, is the weakened seedlings, that is diseased seedlings, they cannot penetrate this 3 mm brick gravels. That is why he used it for seed health testing. The first seed health testing lab, we have seen first seed testing laboratory. That is different. This is seed health testing laboratory. Remember this. This is a government seed testing lab in Wageningen, that is Netherlands, right? And who established it? It is under the chairmanship of Dr. L.C. Doyer, and she is the first official seed pathologist considered in 1918, and she coined the term seed health. Don't confuse with the other term, that is seed pathology. These are the different terms given by different scientists. Seed health is coined by L.C. Doyer, and seed pathology is coined by Paul Neergaard, Mary Nobel, and McDonald. And Paul Neergaard He is the pioneer of seed health testing. That is why we call him as a father of seed pathology. Paul Neergard, the famous book of Paul Neergard is Seed Pathology, okay, where he mentioned what is the significance of seed health testing and various procedures used in the detection of seed-borne diseases. Okay. In seed health testing, the important one is detection is more important, right, than management. If we detect the seed pathogens rightly, we can go for the appropriate management. So in detection, we have a different type of methods. Washing test. Washing test means, for example, we have a smut spore or we have any nematodes. We can directly say, right, in ergot of bajra and all. If we see the infected heads of bajra, I'm telling you, the infected seeds, obviously they will float. By washing suspension and all, we can easily get to know the, okay, what type of it. If it is smut, we will see the some black spores. If it is ergot, the floated seeds, that is called as washing test. Or through washing suspension, we are detecting the pathogen. And incubation methods. Very important method are very important and very conventional methods. These are incubation methods. Incubation means we are using a incubator, or we are incubating the seeds. Incubator means we are keeping the seeds in incubator at, usually I'm telling, 20 to 25 degrees Celsius. Here we have a two methods. Okay, one is blotter method, other is agar plate method. In blotter method, it is given by L.C. Dwyer. Okay, 1919. It's a very important method where we are using a sample of 400 seeds. We are incubating it at 20 degrees Celsius for, in general, for 7 days. It may vary from crop to crop also. And this blotter method, why it is very important is these two methods are included in the ISTA recommended tests. Okay, ISTA recommends blotter method and agar plate. It has been mentioned in the ISTA from 1966. Okay, you remember this. Very few methods are recommended by ISTA for seed health. One of the method is blotter paper that has been given by L.C. Dwyer, where we are using 400 seeds and we are incubating at 20 degrees Celsius. Okay. In blotter paper method, why it is very important, we have a modifications of blotter paper. Okay, one is 2,4-D method, other is deep freeze method. In 2,4-D method, we are using 0.2% 2,4-D. Means we are subjecting a seeds on the blotter papers which are moistened with this 0.2% 2,4-D. As we all know, this is a growth retardant, right? 2,4-D, where it will prevent the germination and all, and it will just promote the fungal growth. So if we use a 2,4-D, we are just going to observe the fungal growth, and based on that, based on the mycelial or colony characters, we are going to evaluate what type of pathogen it is. In this case, we can also go for coumarin, which is also a growth retardant, but the disadvantage with this is it will give some bleaching effect on the fungi. That means it is very difficult to evaluate if it gives some bleaching effect, right? We can't see it clearly. That is why we are preferring 0.2% 2,4-D. And this is majorly recommended for Pyricularia varice, which cause rice blast. Okay, to remember this. Wherever it is specifically mentioned for some pathogens, you have to remember. So modification of blotter is 2,4-D and it is for Pyricularia varice. In deep freeze, what happens? The name itself indicates. As usual, at 20 degrees Celsius for 2 to 3 days, we will incubate. What we will do, again, we will go for deep freeze, that is minus 20 degrees Celsius for 24 hours, that is 1 day. Again, we are bringing back to the normal conditions, that is 20 degree with normal UV. Okay. So we are seeing, since fungi and all, they prefer very low temperature, and at a low temperature they propagate very rapidly. So we are checking for that. That is deep freeze. For Phoma species, for Fusarium, we are using this deep freeze method. Just remember, incubation methods are blotter and agar. In blotter, we again have a modifications of blotter method, that is 2,4-D and deep freeze method. Agar plate method, this is also one of the other method which is recommended by ISTA. Okay. In agar plate, as we all know, in agar method means we are using a potato dextrose agar, and then we are keeping the seeds in the incubator, and we are seeing if any fungal growth or any pathogen grows or not. In agar plate also we have two methods. One is hold fast like method or hold fast method, and other is glycol method. So you just remember why it is modified. In case of agar plate, in general we are going for PDA. In hold fast, we are using 1.2% water agar. Okay, 1.2% water. What is this hold fast? Depends. In case of Phoma betae species, Phoma betae, in sugar beet, right? This pathogen will give us some swollen hyphal tips. The hyphae tips, whatever we have the hyphae, the tips are swollen, not pointed like in general. So they look like a hold fast structures. They are called as hold fast structures. Since it is specifically for Phoma betae species, that is why this method is called as hold fast method, and it is for Phoma betae. Remember this. And here we are using, instead of agar, we are using a guaiacol. It is for Pyraclaria veraceae. This is also mentioned specifically for Pyraclaria. If we see any pink halo around any infected seed, if it is infected, we will see the pink halo around that seed. We see that pink halo, then it is a Pyraclaria veraceae. Just remember these two methods in incubation and modifications, and the specific pathogen for which we are using. Okay. And then NaOH soaking methods. Since NaOH will give a very contrasting color between the infected and healthy seeds, we are preferring it. Where we are using? We have already discussed in the previous part also. It is for kernel bunt of wheat and bunt of paddy. Okay. It is caused by Neoacia indica, it is caused by Neoacia horrida, where we are using 0.2% NaOH to detect the pathogen. And other method, that is whole embryo count method. Here also we are using NaOH method, which is 5%. Whole embryo count is for loose mud of wheat, Chilagonida, right? So remember these 2 points, which is very important. It is for kernel bunt of wheat and paddy, and it is, and also for the whole embryo count method. And serological base. Serological base means based on the immunological property, right? In serology means we are just seeing for antigen antibody interactions. That is immunology, where we are having ELISA, where we are having diffusion test, also the agglutination test. Okay. And other than all this, we are also going for seedling growth test, because some fungi or some pathogens especially, they'll express more clearly at particular stage of crop, right? Some fungi will affect at very seedling stage, like for example damping off. Pythium species, if you see, you can't see the symptom at the mature stage, right? So to detect such pathogens, we are going for seedling grow out test. This is about the general detection methods for seed-borne pathogens. we have detection of viruses, right? Through suspension and all, we can't detect the viruses, and through symptoms also it is very difficult. We have biological and serological assays for viruses. Based on the biological, in biological we have two types, based on the biological properties as well as based on the in vitro properties. Biological parameter properties means by seeing the symptoms, some chlorosis, some venations, any leaf curls and all we can say, but it is very difficult to say which virus exactly it is, right? And by seeing the host also, if we can propagate the host, we can maintain it, then if we assess the host, we can say that okay, since some pathogen, some viruses are host specific, we can also go for this method, but it is not that accurate also. And based on the in vitro properties like liv, in this, that is longevity in vitro. Liv means, longevity means based on the storage. For example, we have a tobacco mosaic virus, TMV. For how much time or for how much duration virus have a capacity to infect, right? For example, for TMV it is 1 year. Even though after storage of seeds after 1 year, if seed had TMV or got infected at the crop stage, even though after 1 year it will again cause the same symptoms and same disease. Okay, that is LIV, longevity in vitro. And TIP, TIP is thermal inactivation point. That is at what temperature we can inactivate the viruses. For example, for TMV we have 90 degrees Celsius. So based on this property also we can detect the virus. And DEP, that is dilution end point. Based on the what dilution, if I took any sap from the infected one and if I dilute it, if it can cause the infection in the, it can cause infection in the other healthy plant, that means till that dilution it is having a capacity to cause that, to cause the disease, right? So that is dilution end point. And cryptopathology means based on some symptoms. For example, for Gemini virus, we have studied this in etiology of some pathogens, right? For Gemini virus we are having paired two wheels, right? This is wheel structure, spin wheel we call it. Paired, always if it is paired, by seeing that we can say that it is belong to Gemini virus. If it is having some pin wheels, it is any rod like structures, it is spotty virus. So based on that such cryptopathology also we can detect the viruses, but they are not much accurate since we can't exactly know which virus is causing the disease. We have some serological assay based on the immunological properties like precipitation, agglutination, immunodiffusion, Western blotting method, and ELISA, which is very, very important. Okay, remember. And some based on physicochemical properties like we have PCR. Different variants of PCR are there. Variants like RT-PCR, that is real-time PCR, where RNA can be quantified, and we have bio-PCR, we have nested PCR. Okay, then UV absorption profile. We can also use electron microscopy. We have nucleic acid spot hybridizations, where we can go for double strand RNA analysis also, and array techniques, that is micro and macro array. So based on this, we have some questions here. Which method is preferred for the large scale testing of the seed transmitted viruses? Okay, the answers are growth test, threshing, southern blotting, and northern blotting. Okay, see the options are not much relative. Let's see, for large scale testing of the seed transmitted viruses means usually we can go for field test, right? This through field test we can go for a large scale testing. And in seeds, RNA viruses can be detected by ELISA, dehydrogenase, RT-PCR, and none of the above. Okay, the RNA viruses are usually detected through RT-PCR. It is a real time, especially qRT-PCR. Okay, which seed testing method is used to detect the seed-borne pathogens? They have given germination test, moisture content test, PCR test. It is PCR, right? We also see for the different variants of PCR. What is the purpose of seed health test? It is very important to determine the germination, to detect the seed borne, to determine the moisture. Firstly, to detect the seed borne pathogens, we are going for seed health test. So, the blotter method, as I explained, of seed health testing detects primarily virus, nematode, bacteria, and fungi. So, very important. The blotter test, which is an incubation method, mainly detects the fungi. Okay. And serological method. Serology means where we are using antigen antibody interaction. It is also called as immunological test. Remember this. It will detect the presence of virus in the seed. Which method we can use for detection of viruses? PCR, ELISA, nucleic acid based test, none of these. So, this ELISA test. These are physico-chemical tests. These are also used for virus detection, but these are physico-chemical tests. And the vigor test method, which estimate decarboxylation activity. It is related to vigor. So decarboxylation activity means is CADA test, where it is glutamic acid decarboxylation test. Glutamic, the principle is more the CO2 evolved, more is the respiration, right? More the respiration, more the vigor. That's it. Remember this. So it is CADA test. So other than all these tests, we have some minor tests like ODV test, determination of huskless seeds. Huskless seeds are determined through percentage, especially in paddy. It is recommended, okay? And ODVs, other distinguishable varieties of the same crop, but to distinguish if there is any mixing of other varieties, the ODV is given, it is by number. It is expressed by number. So some mechanical injury test is also minor test where we have, especially for legumes, where we are subjecting these to ferric chloride of 20%. If it is damaged, it will be stained dark. Okay, it's also very important.
Okay, I guess in the session of like 3.5 hours, so we have completed the seed testing part. The seed testing, as thumbnail says, it's seed testing in one shot. And this particular session on the content that we have provided, it's like from scratch to Z. Okay, it is from A to Z, where we have covered and compiled everything and covered it in a very easy and approachable way, where we have discussed each and everything that is important for the exams. So now, which exams this is important and this is useful? So for all agricultural exams, wherever the seed technology is a actually a portion, you can use it. And mostly, since the end is like ASRB or the AS or the ICAR PhD, till that level, from the basic level, we have covered each and everything. So we'll be also giving you the timestamps for the different tests that we have done, and everything will be given in detail. And I also told you, like, if you want this PDF, you can just fill the Google form and you can directly get this PDF. And if you've already done it before, directly go and check your Google Drive link. And if you have any queries, you can directly text us on this WhatsApp number. So if you are preparing for the ICAR PhD, ICAR PG, or the ASRB NET exams, like AgriAddict is the one-stop solution. You can just join us today and enroll our courses and be the next All India topper for any exam that you take. So Parth was asking, is it helpful for the NABARD exam? Yes, like if the syllabus is agriculture, questions can come in any way. So if these sessions are helping you, you can definitely concentrate. But not everything from the class will be important for the competitive exams. Only basic things will be important. So just try to focus on that and you can cover it from here. Okay. And if you want us to continue this series and provide the similar one-shot videos for all the other parts like seed biology, the seed physiology, including within the seed biology, and the seed licensing and certification, seed quality control, and quality control in that seed testing we are completed, and the seed storage, and you have seed marketing. So let me know in the comment section. So if you people like it, then drop a like and drop a heart in the comment section, and just let me know your views on how was the complete marathon. It is the very first time in a YouTube channel that we have done a 3.5 hour marathon live session on the YouTube channel of AgriAddict. So I just want your honest feedback about these sessions. Based on your feedback, we'll definitely try to come up with more and more sessions if this is really helping you. I'll be waiting for the comments from your side. If you have any things to say, let me know in the comments. Each and everyone, if you want this videos to be continued, I want you to express that in the comment section, and also share our video and keep supporting us. Thank you so much you, and I wish you all the very best for your exams. Prepare well, keep preparing, no matter like if the dates are being postponed or you are not aware of the dates. Don't worry, be consistent, try to prepare for this exam. You have to crack it this time. And for all the students who will be watching this video for the upcoming years, no matter like when you are watching it, this will be like one-stop solution for the seed testing part. Thank you.
Okay, thank you so much for your cooperation and listening to us for 3.5 hours. It's a very good experience for us also. AgriAddict loves you all. So keep supporting us, like, share, and subscribe. We will be coming up with the more and more interesting videos like this. So as sir told, if you really want the same sessions for other units of seed tech, so please let us know in our comments. Thank you. Thank you so much. Good night all. Take care.